Yes, I can hear you, Chris, clearly.
Oh, okay,
Yeah.
thank you. Hi, Deepak.
Yeah, hi, Chris. Yeah, everybody's nice and loud and clear.
So since it's 6:30 already, so we can start on that. So I hope Professor Kaul and Dr. Lata will join whenever they'll be available. Okay.
Yeah.
Okay.
So good evening, good afternoon, and good morning to all the panelists, depending on where you are on the globe. And please welcome to this VaiBhaV Summit session on antimicrobial resistance. So the VaiBhaV Summit was organized by the Government of India to bring together scientists of Indian origin residing in India and abroad to have discussions about topics that are relevant to India, problems that India is facing. And in that direction, this session on antimicrobial resistance was organized. So my name is Deepak Nair, and I am at the Regional Center for Biotechnology. I am a structural biologist, and my connection with antimicrobial resistance arises from the work that we do in the lab on error-prone DNA polymerases. So basically, the how genomic variation arises to give rise to drug resistance is one of the focus areas in the lab. Now, to start with, I will call out the name of each panelist, and you can give a brief introduction about yourself. So I'll start with Dr. Babatosh, who's the co-organizer and one of the moderators for this panel discussion. Dr. Babatosh.
Thanks, Dr. Nair. So hi, so my name is Babatos, and I am working in Translational Health Science and Technology Institutes almost in the past 8 years. So my labs is mainly work on the genomics. So we are mainly working on the antimicrobial resistance on escape pathogens, and we have also started working on the metagenomics in last 5 or 6 years. So the initially idea to just do the metagenomics to have idea what is the resistant gene pool, those are living the human associated bacteria. Then we are isolating each of those isolate, doing their individual genome sequence to identify what are the different resistant genes that do exist, and whether these resistant genes are part of the core genomes or they are associated with mobile genetic elements. And if they're associated with mobile genetic elements, then what are the functions that basically modulate their transmissions among different bacteria. So with that, I will end here, and I'll request next Professor Ramaswamy to introduce yourselves.
Hi, my name is Ramaswamy. I work at Purdue University in the Department of Biological Sciences and Biomedical Engineering. I just moved here a year and a half ago. Before that, I spent 10 years in Bangalore. Before that, I spent 10 years at the University of Iowa Medical School, where my journey into antimicrobial resistance started. My lab initially got interested in toll-like receptors. We identified the ligand for toll-like receptor 4. That was really long ago. A lot of work has been done in toll-like receptor 4. But in the last 10, 12 years, my lab has been interested in how commensal pathogens uptake Nuc5AC, which is the sugar which our immune system recognizes as self and coats themselves with it, and then becomes kind of steals to the immune system and causes sepsis and kills. We have published over 25 papers in this area. We have figured out the pathway. We have done the structural function studies of all of these. And in the current phase where we are is we have now shortlisted 2 or 3 of these as possible targets and beginning to do the drug discovery aspects of it. But we're interested in many other aspects of antimicrobial resistance. I also I've been very deeply involved in doing startup companies that work in this area of antimicrobial resistance, protection against pathogens and stuff like that. So I'll stop there.
Thank you, Rams. Dr. Anil Kaul, please introduce yourself. Hello.
Dr. Call,
Hello.
you muted. You, I think, need to be unmute yourself.
Yeah.
Yeah, can you hear me now?
Yes, yes. Very loud and clear. Yes.
Okay, Dr. Das, thank you. And good morning, good afternoon, good evening to everybody. My name is Anil Kaul, and I'm currently working with Johnson & Johnson. I'm heading their global public health and infectious diseases R&D across the world. I'm an infectious diseases scientist, and I've been in infectious diseases biology for last several years, especially TB and several other infectious diseases, both viral infections as well as bacterial infections. And I was, till last year, I was also director of an institute in India, which is called CSIR IMTech. It's an institute part of the government of India based in Chandigarh. So I headed that particular institute. And now I'm back, as I said, in the global public health R&D, mostly in translational discovery, translational medicine, and I'm also associated as an adjunct faculty where I have a lab in London School of Tropical Health and Medicine. So it's an appointment which happened this month alone. So we are trying to set up a new lab in that particular institution. So it's nice seeing you all, everybody here. It's wonderful being part of VaiBhaV. So let's see how far we can carry it further and know each other a little bit more.
Thank you. Thank you, Dr. Kaul. Dr. Ankur Mutreja, can you introduce yourself, please?
Yeah, hi everyone.
Hi.
First of all, thank you for having me in this forum. It's very nice to connect with all the scientists in the infectious disease space and AMR in particular. So I'm currently based at the University of Cambridge in the Department of Medicine. I lead a global health group here that particularly focuses on the transmission and spread of diseases, and using genomics, we go ahead and use that data to develop new diagnostics and vaccine candidates. Prior to coming to the University of Cambridge, I worked in industry. So I worked at the Hellman Laboratories, that is a Wellcome and Merck partnership company, developing ETEC, cholera, Shigella, typhoid, and rotavirus and meninge vaccines. And before that, I worked at Novartis, where I was involved in development of Group A streptococcus vaccines. So my broad interest is very much in the infectious disease space and AMR space, and therefore I'm very happy to be participating in here. Thanks very much.
Thank you, Dr. Ankur. Dr. Saurav Ghosh. Hello.
Hi, everyone.
Hi.
Glad to be invited to this forum. As you can see, it's excellent weather here, and I'm really enjoying. I'm currently based in Loughborough University. I have a research group focusing on rapid diagnostic test development for biomolecules and microbes. Infectious disease is one of my key areas of interest. I'm a mechanical engineer by background, and then I specialized in biomedical engineering when I was in Oxford. But then from there on, I started my journey of development of principles, mechanisms, and assays and devices around rapid detection of infection. So I did my PhD in Cambridge, and also my postdoc during that era. I was looking into acoustic techniques, so how vibration of small electromechanical oscillators can be used for rapid detection of bacteria. And then gradually the antimicrobial resistance wave started. So then we started worrying about, is detection of bacteria good enough, or do we need to do something more? And then over the last 7-8 years I'm in Loughborough, and we have been looking at both the nonlinear acoustic technique that we developed, and we also developed a sensor out of that, which is now used in quite a few universities. And we are also looking at single-step fluorescence-based detection or colorimetric detection, which allows a rapid within 10-minute detection of both the presence of bacteria, that is a bacterial load, as well as their antibiotic susceptibility or sensitivity. So that's what we are focusing on, and we have worked quite a bit on urinary tract infection tests, also influenza, and that is what has led us to the SARS-CoV-2 as well, but that's another story, I think.
Thank you. Dr. Manoj?
Hi, yeah, thank you so much for inviting me on this forum. My name is Manoj, and currently I'm based at National Institute of Health as a postdoctoral scientist under working for Dr. Shankar Adya. Before I came here, I did my PhD from Texas A&M University, and my PhD work was primarily focused on studying bacteriophages, and in particular, understanding the lysis process. How do the bacteriophages cause lysis of the gram-negative bacteria, especially in particular, understanding the disruption of the outer membrane of that, and working in the paradigm phase lambda, that I'm sure you all have at some point have heard about it, have knowledge about it. And from there, while I was doing my PhD in phase work, and this, as we all know, the antimicrobial resistance wave have been going on and constantly rising. So I got interested in still working on the same field, and phase therapy was kind of getting an excitement and a hype here, and I still continued with that and came and joined the Dr. Shankar Adya's lab, where we are currently isolating different phases from the environment and characterizing them. And also we do multiple different types of work, such as genetic engineering of the phases to target, different pathogens or different hosts, just to, broaden the host range, things like that. And I'm really glad to be in this forum, and I hope I can contribute something from the phase perspective, how it can be utilized to mitigate the problem of the antimicrobial resistance crisis. Thank you.
Thank you. Dr. Sulagna Vasu?
Can you hear me?
Hello?
Yeah, can you hear me?
Yes, clearly, yes.
So, first of all, nice to see a lot of people here across the globe, across different time zones, who are interested in antimicrobial resistance. It's nice to, I have known some of you, and it's nice to be knowing others, new scientists here. And I am from the National Institute of Cholera and Enteric Diseases. So primarily, I've been working for the last 15, 16 years on antimicrobial resistance and focusing on neonatal infections. And slowly we are moving into other infections too. But in the neonatal infections, we look into the bacteria that primarily cause sepsis in neonates, which is Klebsiella pneumoniae and Acinetobacter. Again, focusing on carbapenem resistance in Klebsiella, E. coli, Acinetobacter, because that itself has become a real problem, particularly in the developing world. We also look into the gut microflora, the neonatal gut flora, and antibiotic resistance against again looking into the resistant genes in the flora, and we primarily focus on the transmissible mechanisms, but we are also, for Klebsiella pneumoniae, we are also looking into the other efflux mechanisms also. So, so that's how I've been doing this for a long time now. So it would be nice to hear you all and to see what I can learn from each of you here.
Thank you. Dr. Sumant Kandra.
Hello, and thank you for inviting me. And I'm Sumant Kandra. I'm an infectious disease physician, an adult infectious disease physician, and a public health researcher. My work mainly focuses on understanding the drivers of antibiotic use on the human side mainly, and also looking at hospital-acquired infections and how to prevent those infections. Currently, my research is funded by CDC here, and we're looking into understanding the burden of multi-drug resistant bacterial infections in Indian hospital. And the goal is to come up to see, is there any way to prevent those infections? And I was also part of the scoping report that was prepared to the Department of Biotechnology a couple of years ago. And I've been on the WHO expert committee on antibiotic use since 2017. And I'm looking forward to work with you guys and see where we can collaborate in the future.
Great, thank you. Dr. Animesh Rai.
Yeah, greetings everybody. I'm Dr. Animesh Rai. So I'm based out of New Delhi, where I work as a consultant in the Department of Medicine at All India Institute of Medical Sciences, New Delhi. Now, my department, Department of Medicine, it runs its own infectious disease clinic, which is first of its kind in the country. Besides catering to OPW patients as well as having its own intensive care unit, my department also has started the first IED clinic course, the DM infectious disease, which is again the first in the country. So my association with antimicrobial resistance is mainly due to my clinical practice. Antimicrobial resistance is a common problem in the kind of practice which we do at Department of Medicine at All India Institute of Medical Sciences. And my research areas, among others, include pulmonary infections. I look forward to an evening of discussion and learning. So great to be here. Thank you.
Thank you. Dr. Sayantan Banerjee.
Good evening, everybody. Good morning to my friends in the West. I hope I'm audible, loud and clear, right?
Yeah. Yes.
So thank you for inviting me here. I'm Sayantan. I'm basically an infectious disease physician. I did my postdoctoral studies from the All India Institute of Medical Sciences, New Delhi. And after that, I have been the focal point technical officer for antimicrobial resistance in the World Health Organization Southeast Asia Regional Office, which I have quit very recently, 1 month back. And now, basically, I am working for an international nonprofit named One Day Sooner, in which we basically have developed cutting-edge protocols, ethical standards, and advocacy campaigns for fast tracking of COVID-19 vaccines. So since January this year, I have also been looking after the Rapid Research and Innovation Cell for COVID-19 in WHO CRO. And as focus has shifted from everything to COVID-19 in the last few months, in fact, a lot of our AMR works in WHO has actually shifted interest towards COVID-19. In fact, we have started working on co-infections in COVID-19, and we are facing a significant burden of AMR in COVID-19 patients who are actually being mechanically ventilated and treated in the ICUs. We can have a separate discussion on that. And basically, currently, I am working on human challenge trial protocols for rapid approval and equitable access for COVID-19 vaccines. My postdoctoral research basically was on colistin resistance bacteria. And we have seen a very strange phenomenon. We are about to publish this, is when we have long-standing patients in our indoor patient, in our IPD units, and they have been treated for systemic infections by parenteral colistin or polymyxin B, within 7 days, half of them, and within 2 weeks, almost 3/4 of them develop colistin-resistant colonization or by colistin-resistant enterobacteria in their gut flora, which is a work jointly done with THSTI. Here I have Dr. Bhabatosh Das, who was my OPI in this work, and I have been personally interested in AMR due to my practice as well as my role in WHO, and looking forward to learn from you and participate in rich discussion today.
Thank you. Dr. Krishna Mohan.
Good morning, good afternoon, and good evening to all of you. Thank you to Bhabatosh and Deepak for organizing this and making me part of this particular forum meeting. It's a great opportunity to interact with all of you. So my lab is involved in looking at understanding host pathogen interactions by using Mycobacterium tuberculosis and Acinetobacter as model systems. So we use a variety of techniques to understand how these pathogens are able to modulate the host cellular processes by delivering their own arsenal, and how this particular arsenal is able to co-opt and subjugate the host cellular processes to develop a niche. And in the process, we end up having discovery of several unique novel targets, which we use either for diagnostics or as vaccine candidates or as therapeutic targets. So my interest with AMR is because of my collaboration for the last 6 to 8 years with the AIMS neonatal faculty in neonatal sepsis. And so both in MTB as well as with Acinetobacter, we are trying to look for alternative ways in order to reduce the emergence of the antibiotic resistance. And one of the ways we are looking at is whether if you were to target therapeutically some of the most virulent genes of these pathogens, is there a possibility of attenuating the pathogen so as to reduce the total load of the antibiotic delivery into the patient, and whether that would be able to reduce the dose of the antibiotic, reduce the treatment duration, and possibly leads to the elimination and loss of persistence. So this is the reason why I'm part of this panel. Also, in the recent past, we have had a lot of interest in the neonatal sepsis. So my lab is looking at, together with Bhabatosh, we are looking at the transmission dynamics of some of these escape pathogens and trying to understand whether these pathogens are coming from the mother or from the hospital environment, especially to cause the early onset sepsis. And in addition to that, we're also looking at the genetic variations in collaboration with IGIB, looking at the genetic variations of the host that possibly dictate who becomes susceptible to the neonatal sepsis very early on. And the other thing is also that we're looking at host responses to these particular pathogens to see whether you can utilize the expression dynamics of a subset of these particular host proteins as a way, or a transcriptome, to look for whether we can identify the sepsis infections much early on as compared to even seeing the symptoms or the possibility of the cultures becoming culture positive. Thank you all, and I look forward to a great interaction.
Thank you. Avinash?
Hi everyone, I'm Avinash Bajaj. I am here Associate Professor at the Regional Center for Biotechnology. I'm here from last nearly 10 years. So quite different from each one of you. I am trained as a chemist, and from IISc Bangalore, I did my PhD. And for the last 10 years here, we are working on different molecular scaffolds where we synthesize different chemical entities. One is the area of drug delivery, and other is to target the different bacterial membranes to make them more specific. Can we have a specific, for example, for the mycobacteria or any for the candida? So in that journey from last 7-8 years, we have done some achievement. For example, in case of diagnostics, the challenge is to differentiate between extrapulmonary TB in case of Crohn's disease as well as the gastrointestinal TB. So we have synthesized a probe which can specifically detect the mycobacteria in the gastrointestinal tissue. So this was done with Dr. Vineet Ahuja at AIIMS. And apart from that, we have done physicochemical parameter standardization, where we know what are the features which are needed in the small molecules which can specifically bind with the LPS, for example, to target the gram-negative bacteria. So now we are working more on those scaffolds to make a library of around 1,000 molecules which can potentiate the existing especially the antibiotics which are used basically for gram-positive bacteria, how we can use them for the gram-negative bacteria using these molecules so that you can enhance the drug accumulation in gram-negative bacteria. So these are our interests. So looking forward to this discussion. Thank you.
Thank you, Avinash. So all the introductions are over. Dr. Lata Kapoor, who was supposed to be part of this discussion, could not attend due to some personal reasons. And Dr. Bhavtosh has made a list of topics and questions within those topics. So he will go through each of those topics one by one, and we can have discussions. There are some names associated with each topic, but everybody can give in their opinion about these topics. So over to Bhavtosh. Dr. Bhavtosh.
Thanks, Dr. Nair, for briefly introducing these sessions. So what actually we have structured these sessions in 6 different topics. So each of the topics will have around 2 or 3 questions, and the relevant expert could discuss about those different questions. We have also activated the chat box in the YouTubes for the audience. If is there any specific relevant question, so we'll pass those questions to the panelists for discussion. Okay. So the first sessions, what we made, that's based on the discovery research and development of new antibiotics. So we know that the antibiotic is one of the most crucial drugs that developed in the medicinal history. It saves trillions of lives, and it also increased the life expectancy of the people's more than 20 to 25 years. But since its discovery or introductions, it was observed that the antibiotic have 2 effects. The one that is the desired effects, it's kill or cure the infections or prevent the infections. But the other undesired one, it's basically promote the proliferation and spread of antimicrobial resistant pathogens. Since it's kill the susceptible population, the definitely resistant population, they have resource, they have environment to multiply, and that's the major problem. So this antibiotic resistant bugs, that is not human beings. This is the natural phenomenon, and it exists like millions of years ago. So before we go to the antibiotic resistance, the first things what we feel really need to know. So what are the current status of the discovery of different antibiotics or drugs or biologicals? And what are the major challenges in the antibiotic discovery research? I think we should start with Professor Anil Kaul, because I think one of the most renowned scientists who is working in this world on the discovery and antibiotics, and who has also invented the last line drugs of the bedaquiline. So I request first Professor Kaul to at least give us his thought regarding the current thought of the antibiotic resistance research and also the challenges in antibiotic resistance. Professor Kaul.
Yeah, thanks, Bhavtoji, and thank you very much. And I think this is a very nice panel. You have a very assembled, very nice set of people who have diverse experience. So, from the discussion perspective, it will be very nice to see how both from the clinical side as well as the discovery side as well as the medicinal chemistry side, how people see AMR in general. But I can tell you that we are living in COVID times, which is basically unprecedented. We never thought that the world will come to such a state, what we are experiencing right now. And this is basically due to a viral infection. And now, but I'm sure that if the world goes the way it is going right now, we will experience a big, huge challenge with AMR. AMR is going to be the next pandemic if things stay the same as they are right now. And all of you know the projections from WHO in terms of the impact on the economy by 2050, the famous slide of WHO. People must have seen how AMR is going to change the world and impact the economy. And particularly India, I'm more concerned about India, because India, we have a special, more graver threat with the AMR. I think we have in Europe. Europe is still managing the AMR by proper hospital controls, like MRSA was a big, huge issue in early 2000 here. A lot of countries, but they did manage to control the MRSA spread and increase the resistance of staphylococci. But what we have seen in India is a different trend. I think there was somebody in a panel who has worked on the colistin resistance. And when I was in MTech, we have seen in the PGI a lot of people who had who were administered colistin, but eventually even with the colistin treatment, they had the resistance to colistin. So we know Pseudomonas on Acinetobacter resistant colistin is going to be a next big issue in terms of the hospital-acquired infections in India. So if you can imagine, the fluoroquinolone resistance in India in some cases goes to 70%. So the major drugs what we have in India in terms of the treating infections, especially the fluoroquinolones, they are losing their efficacy. And that's a very scary part. I think that's the most scary part. I feel that hospital-acquired infections, particularly in major hospitals like PGI, like AIMS or other places, it's really serious. It's really serious because we have monitored, we have data now from hospitals across India, and we see Acinetobacter baumannii as a big, huge challenge. We see Pseudomonas as a big, major challenge. We see gram-negative infections as a bigger challenge. So, so from that perspective, I think we as a group, as a nation, as a country, we need to do whatever it takes: proper diagnostics, proper surveillance, proper health control measures, whatever it needs, and new drugs. And we definitely need new drugs. So, so unfortunately, the industry has not been very much interested in the AMR. There are no new drugs which are being developed. And of course, and those drugs which are being developed cannot reach to the right patient population because there is no market for them. There is it's not only discovering and developing drugs. That's one aspect of it. Getting drugs to the right patient population is much bigger challenge than each one of us will understand here. So the supply chain of antibiotics is really, really disrupted because of it's mainly driven by the fact that return of investment in antibiotics is very, very low. If you are going to invest $500 million or $1 billion, and return is less than 100 million or 200 million, nobody is going to bring that investment. That's why most of the antibiotic research right now is really focused on we need to generate new models of looking at investing in the antibiotics, particularly the AMR. And I don't know whether you have heard about it. There's a $1 billion fund which was recently created. I think it was a news of last month, where this $1 billion is created with the aim that by 2030, we will have at least 2 or 3 antibiotics, which will be in development. And then we have to identify mechanisms through WHO and other partners which can distribute those antibiotics, as we have done with Bedeclin. So when we started with Bedeclin, particularly my own story, we started with a project where we knew that there was no return. It's not a commercial venture. It's an TB kills mostly the poor man, and there's no money to be made. But I think what we could do is that we did some very interesting science. We did discovered a new target. We discovered a new chemistry. We discovered new biology, and we also developed new clinical trial protocols where nobody had done any clinical trials in the last 50 years. So from that perspective, we did something which was really unique and innovative. And from that innovation came Bedeclin, which is right now one of the most promising drugs in TB and MDR TB space across the world. It's used in more than 140 countries. It has gone till now to 200,000 patients. So there is an immense impact of antibiotics on the lives of people. I was in UP in a hospital in BHU, and I can give you an example. It's a very interesting example because what I saw when I was there for a talk or something. So in that particular hospital, they had an MDR TB center. So in that MDR TB center, there is Professor Srivastava. He's an amazing physician, fantastic guy. Has been treating TB patients for last 30, 40 years. So when we met, he said that, let me take you to the MDR, show you one patient. So there was one girl, she was around 21, 22 years old. She was on the hospital bed. So and he said that this girl is being treated with Bedeclin. So that's fantastic. That's so nice. So the government of India had approved Bedeclin for MDR treatment across all over India just 1 month before. But she said that, yeah, it's a good story, but it's not that really good because her husband died of MDR TB or XDR TB. Her older daughter died of XDR TB. The mother had, the lady had XDR TB, and her younger daughter was infected with XDR TB. Only the small son, which was 4 years old, was 5 or 6 years old, was not infected at all. So you can see there are certain regions in India, especially Purvanchal, how much impact drug resistance, whether it is TB or whether it is Pseudomonas, whether it's Acinetobacter, is having. And we know the impact of TB because the government is very keen to have a big, huge impact on TB, but the resistance emergence for Pseudomonas and Acinetobacter is a big, huge worry for me. So yeah, this is I'm just trying to make you guys all understand. I think you all are experts. You know how challenging it is to develop drugs, to bring it to the markets, to develop new innovations in AMR space, but it is desperately needed. We really need to do something right now to save lives in future. We will be otherwise in a worse situation than what COVID has created. I don't know whether that was the right answer, but it was a long answer for a small question, but I just tried to frame the issue which is in front of us as a group.
Thank you, Dr. Kaul. So our next question to Dr. Ramaswamy, that we know that a lot of antibiotic discovery is going on, but when we look exactly when a new antibiotics come, so most of the times it's claimed that it's just a modification of the existing drugs. So we could find hardly any antibiotics with the novel scaffold. So sir, could you please explain us that why new scaffold is not coming? Why we are only concentrating on the existing drugs? Because if we modify the existing scaffold, automatically resistance will also come quickly.
Thank you. Let me put things in a little larger perspective, right? So there are 41 new antibiotics in development, both between industry and academia. These drugs would potentially address many, but not all resistant bacteria, okay? Not all of them. We've gone from the idea of making very general molecules that will kill a large number of bacteria to being a little more specialized in terms of very specific groups of bacteria. So that is a change in the mindset. And hopefully the change in the mindset is what is required in order to find new targets that are very specific. Because if you look at all of the existing targets, they were all gyrations or ribosomes, which are generic among all of the large class of bacteria. So as we begin to narrow the groups and make specific antibiotics, hopefully our targets will increase, okay? However, given the inevitability that some of these antibiotics will fail to win approval because there are 41 in development, and that resistance will eventually develop to those that are already approved among this 41, it's clear that there are too few drugs in development to meet current and anticipated patient needs. This is December 2019 data, only 41. Of the 41 antibiotics in development, 15 are in phase 1, 12 are in phase 2, 13 are in phase 3. One has had a new drug application submitted, 4 have been approved. History tells us about 60% of the drugs in phase 3 will be approved. So if you take now the number in phase 3 is 13, about 60% will be approved. So we will have about 7 new drugs in the next year or two. At least 18 antibiotics in clinical development have a potential to treat infections caused by gram-negative escape pathogens. Dr. Anil Kaul said the importance of gram-negative bacteria. So about 18 of them are against gram-negative escape pathogens in a critical area, right? So that we just heard from Dr. Kaul. There are at least 10 antibiotics, if approved, could address infections caused by drug-resistant bugs like Neisseria and the Clostridium family. So there is a whole problem there. But these are, again, as one of the panelists said, one of the most urgent threat of public health are the Clostridium family. Approximately 1 in 4 drugs in the pipeline represent a novel class, which means now out of the 41, only 1 in 4. So we have 10 new novel class of drugs. And none of these novel class of drugs are in the phase 3. So we don't know how they will fare as we go along. And unfortunately, none of these novel class of drugs are against gram-negative bacteria. So I'm just providing a large amount of data, and I'm happy to share this data with people as well. And there are about 35 or so companies with antibiotics in clinical development. All of them, as Dr. Kaul said, are not the big players. They're all the small players. And there are only one of them, for example, of these 35, ranks among the top largest 50 pharmaceutical companies, which means for reasons Dr. Kaul said, the big players are not interested in this. So we are left startups to actually take majority of the load of creating new antibiotics. I mean, so that's where I'm involved in doing a bunch of stuff. So to answer your question specifically, 90% of the molecules among this 41 are modified ones, which means resistance will come quickly. But there is about 10% that are novel, and the novel ones are beginning to come, but they are not going to be this antibiotics which will address a large number of bugs. There will be antibiotics that will be, say, specific to the Clostridium family or specific to this, which essentially means we will have to develop new tools to very quickly find out what type of infection you have and just not say you have a bacterial infection. I'll stop there and answer any questions. I didn't talk about my research, but that's a different issue on trying to develop new targets. But this is where the status is, and this is the reason why we don't see many new class of antibiotics.
Thank you, sir. It's really very nice summary. So now our next question to Professor Bajaj. So since Professor Bajaj is working one of these country premier institute, and he involves in the new antibiotic discovery. So we just need to know in Indian perspective, what are the major drugs came in the last 50 years from academy and industry? What contribution India made in the development of new antibiotics? If there is not substantial contribution, then why?
So thank you, Bhavtosh. I think Dr. Cole and Dr. Ram has set the platform and make it easy for me to compare. So as Dr. Ram said, he has mentioned about what are there in the different clinical phase 1, phase 2, phase 3 clinical trials. Now, if you see in just a few months back, the review in Nature, Review Microbiology, they have basically compiled all over the world data about the preclinical studies. So you need to see what is happening in the preclinical, which has some scope to come to the clinical. Now they compared basically the 4 big fundings, which we all know: Carbax, Repair, Enable, and JPAMR, right? So when they compared basically, if you see that there are 300 institutes who got these grants, and in most of them 136 was in USA, and in India there are only 3, or you can say either academic institutes or the small companies who are working on this antimicrobial new drugs, antibiotics. And among these institutes, if you see globally, there are only 10% which are academic institutes, no big companies, only 3%. And there are major, what Dr. Ram said is that small companies, R&Ds, which are startups, which are major part is 80%. Now, very interestingly, if you see the preclinical things which are going on, the 40 to 50% are the projects which they are targeting the small molecules with a new target. So now, very interestingly, if you see the compare between the preclinical as well as the clinical data, as Ram mentioned, there are 41 antibiotics in the different phases. Now, if you see 70% of the proposal in the preclinicals are finding the new targets. Whereas if you go to the clinical studies, only 90%, 19%, 20% are, 25% is on the new studies. So from preclinical to clinical, you basically see the new drug molecules, new targets don't have that much scope because they fail, because they are totally new, nobody knows. Whereas the old drugs, 75% are in the clinical trials. So this is so when we need to design the molecules, we need to think about these things to see what is there in the clinical trial. So we need to refill this antibiotic pipeline to see how to get a broad spectrum. For example, as mentioned, different gram-negative bacteria is a major challenge. How to find a broad spectrum antibiotic which need to be bactericidal, and you need to see different drug resistance mechanisms, not only what the bacteria generate themselves, but there are persisters, there are biofilms, right? So need to find that. So in India, as I mentioned, that there are only 8 projects out of the 400 projects which have come to India. So this is one of the, I think, need to be addressed.
We received one question from the audience and from M. Sengupta. So what she wrote that, what are the prospects of repurposing existing non-antibiotic drugs? So could either of you please would like to answer these questions?
Yeah.
So go ahead, Avinash. Avinash or Anil are better to answer that question than me.
So basically, if you see in the preclinical things which I was discussing the data, not that many projects have been funded by different agencies, very interestingly, everybody is looking for the new targets, new antibiotics, although they fail in the clinical trial. But repurposed drugs has a much bright future. But if you see, only 4% among the projects are on the repurposed drugs. So, but from the last few years, the DBT has taken that initiative, and especially have pushed the scientists to start this. So this has just started around 2 years back. So we hope that we may, from Indian perspective, we may get some new hits which can be from the repurposing of drugs can be used in future for the clinical application.
I think people, it's not a new idea to repurpose drugs for antibiotics use. People have tried repurposing drugs for last 20, 30 years. Means we all know FDA approved drugs, they are all there. We all know these drugs, and people have screened and these 5,000-10,000 library FDA approved drug library several times. It goes several different pathogens. So the data which has come out of those is that these are not really effective antibacterials. They are most host directed, but as direct antibacterials, they are not very effective. So that data is clear. I think this constant repetition that we should be looking at the repurposed drugs, it will yield us more new molecules. That's not really true. Yes, of course, what we don't understand is that whether we should look at new drugs which target the host rather than the pathogen. So it has happened historically very well in viral diseases. Like if you look at hepatitis C was very effectively managed by interferon therapy. So, but now once again in hepatitis C, you have all direct acting antivirals in that space. So, in HIV, people are looking at PD-1, all the checkpoint inhibitors, where you direct the host or T cells, modulate the T cell response to a particular pathogen. So, but these are all new. Host-directed therapy is still a very, very far-fetched idea because we don't understand the biology in terms of impacting the host and thus as such killing the bacterium or the virus. So for the moment, I think repurposing will be a long shot if we are looking at direct antibacterials. But what we need is new chemistries. We need new natural scaffolds. We need new targets. We need new biology. So that's what is missing basically. And whether we have a narrow spectrum antibiotics or broad spectrum antibiotics, whether we can use microbiome, the host microbiome in terms of modulating, having an antimicrobial response. So there are several new things that need to be defined where our understanding is very, very limited. So the metabolites from the microbiome, which we all have, how effective they are, they can be in terms of generating new scaffolds as new antimicrobials. So there are several interesting ideas, but the challenge for most of the antibiotic research, as was said earlier, is basically the lack of incentives to drive the whole area forward. So I think unless and until you incentivize the R&D, you increase the budget, it's not only discovering the target, it's not discovering the chemistry, it's enabling that discovery to eventually reach that patients. So that long haul from the molecule to the patient, it takes millions and millions of dollars. How do you secure that funding line in order to bring these molecules to the patients? People are trying it now, and that's why advocating public-private partnership is key here. And we have done it in TB. TB is a classic example where you see the TB pipeline now today is very, very rich. And if you look at 15 years ago, the whole TB pipeline was completely dry. There was nothing new in there. Now we have 3 or 4 new TB molecules getting approved in the last 3-4 years. So there is a lot of momentum, but this only shows you that if there is a purpose, very defined clarity in the vision and the funding, I think things improve. And the same thing is with antibiotics. Of course, we are not talking about vaccines here right now, which is another part of the whole picture. But overall, I think repurposing will not really serve the purpose.
Dr. Kaul, I have 3 questions for you, and maybe this is what, to me, the take-home message of this conversation is that Deepak and his colleagues should take back to the government. You said 4 things, which are hit the nail. One is we need new targets, which is investing in basic science and understanding. Our understanding is limited. Therefore, a significant amount of investment has to go into basic biology. Now, we don't want to invest in basic biology, right? We always want to translate, and we have nothing to translate. Translation, the word means I have something in one language, I'm going to put it in another language. In the absence of any understanding of the biology of infectious disease, there's going to be. So we need investment in new targets, which is basic biology. We need investment in new biology itself of the understanding of the disease, and we need new scaffolds, which is investment in chemistry. Now, the message that one needs to take is that this is a long haul, as you said, and the investment in the basic biology has to significantly go up. DBT's budget today is $300 million US. I mean, that's less than the most American universities, the top 50 American universities have for their biology program for the whole country. So if India is serious about tackling this AMR problem or any problem, and AMR would be a problem that India should focus on rather than cancer or neurodegenerative disease, because this is a problem that's going to kill more Indians than any other disease, why don't we take DBT's budget to invest, DBT, ICMR budgets to the 2% of the GDP and invest $5 billion into this, instead of investing $300 million and most of it goes away in feeding its own institutes and not really funding research. The idea that somehow we can do translational research without basic biology, and you clearly articulated why this is important, is amazing. So I think that's the message that I think the top group has to send to our administrators and the Prime Minister's office, or whoever is listening. If they want us to do this, let them listen to us.
Thank you, Professor Ramaswamy. It's really a nice message to the funding agency as well as the administrator. I also agree that until we invest money on the basic or fundamental research, nothing will come in translational aspect, because we translate where we have our basic understanding. So now we are moving to our second section. So antibiotic resistance is a natural phenomenon, and it exists, I think it's thousands of years ago. So question is that, so what are the major source of those antibiotic resistant genes, and how to track the emergence and spread of antimicrobial resistance bacterial pathogen? So in last 10 years, there is a huge development of the DNA sequencing platform, and due to the availability of the DNA sequencing platforms, thousands of pathogenic DNA sequences available in the database, and one of the best way to track those antimicrobial resistant genes reservoirs in the bacteria by completely sequencing their genome. So we have one excellent expert who is working on the bacterial genomics, I think in last 10 years, and have several contributions. So I'll request Dr. Ankur, just if he can explain how to track those resistance, emergence of resistance, and also to identify what are the major source of antibiotic resistant genes in human associated microbiome, as well as in the environmental condition. That will definitely help the audience. Ankur, please.
Thank you, Bhabatosh. I agree that genomics has now been there for quite some time, but despite that, the efforts to track antimicrobial resistance has actually not touched the pin that it should. Thinking about how to track the spread of antimicrobial resistance, I think before that we have to think how to track the spread of bugs that are actually very, very good in carrying these resistance genes, or the mechanisms through which they become resistant. So we have seen, and so have others as well, that there are bugs that are incredible at picking these resistance genes, which not just help them for their own survival, but they also help these bugs to spread faster, to cause more severe disease, and increase their transmission potential. We've seen this repeatedly in cholera. We've seen this in typhoid. We've seen this in non-invasive, invasive non-typhoidal salmonella, and other bugs such as Klebsiella and Acinetobacter, which are hugely problematic from hospital-acquired infection point of view as well. And tracking those bugs takes us to genomics, where there are areas in the genome that can actually give a lot of information about what actually makes these bugs very good at first picking these antimicrobial resistance islands or cassettes, and then sharing them with their friends, which could be part of the microbiota, whether that is gut microbiota or respiratory tract microbiota. So I think it's very clear now that there are several lineages that people have to track and understand the mechanism by which these lineages have become so successful in picking these antimicrobial resistances from all sorts of regions, whether that is environment, whether that is natural, whether that is actually from the commensal source, or whether that is from the heavily insecticide infested food that we eat these days. There have been several studies that have shown that actually if you eat a couple of spoonfuls of honey that is commercially available in the market, you actually end up taking more than 5-fold the dose of antibiotic that you would be given if you had a normal uncharacterized febrile illness. So there is clearly that area that needs to be addressed. Fishery is a big area, poultry is a big area, all the meat industry and vegetation, everywhere efforts have to be coherent. And then comes the side of host and pathogen, where we have to understand the dynamics of interaction, why these bugs, despite sometimes being in environments where they actually would not need antibiotics, would not have any sort of selection pressure, why would they still have these pathways that would eventually end up giving them the antibiotic resistance? So I think in terms of tracking, genomics is definitely the way forward. Everybody has accepted that now as a gold standard, including WHO, CDC, and all the major public health agencies, and including VaiBhaV, and in collaboration with VaiBhaV and others of this forum. And in India, we've done several studies now that showcase that we can effectively track antimicrobial resistance, identify the lineages that are very heavy in AMR, and then potentially try and target those lineages through all kind of mechanisms, whether that is very narrow spectrum antibiotics or using antibodies targeting those particular lineages, whether those are O antigen targeting antibodies or LPS targeting. And then also exploring the area of vaccines that ensure that the candidates in the vaccine are now covering the AMR as well. And there are several examples in enteric disease space where the attention is now being focused. So I hope that answers that question, VaiBhaV.
Yes, Ankur, thank you. So since from the genomics, we could easily understand that this antibiotic resistance could be due to the accumulations of mutations, or it could be due to the acquisitions of novel function. So when particularly a antibiotic resistant genes acquired by bacteria, that give major problem to the antibiotic what we call the industry, because they could disseminate very fast. So we have a very well-known scientist, Dr. Krishna Mohan Atmakuri, who is working in THSTI on the horizontal gene transport, I think more than 15 or 16 years. And Chris is the best person who can explain how those antibiotic genes that link with the mobile genetic element would disseminate among bacterial population. So Chris, please over to you.
Thank you, Baptosh, for the lovely question. To a large extent, in this era, the antibiotic concentration in the environment has been dictating the emergence, the selection, the distribution, and the maintenance of the resistance in a bacterial population. So in general, you can think of possibly two scenarios wherein you have the presence of the antibiotics dictating how the selection and the distribution happens. And the other is in the absence of the antibiotics, what exactly happens in this particular population that leads to the resistance continuing within these populations as well as maintaining and widely distributing. So as we know, in the presence of the antibiotics, the phenomenon is straightforward. There's nothing much to understand, but to say that as the antibiotic concentration becomes very high, the susceptible pathogen basically gets dead, while it leads to the selection of the resistant pathogen. And the growth of them will be determined on the fitness cost of this particular resistance that brings into this particular population. Also, the sublethal doses of the antibiotics in the environment has been shown to be largely influencing the dynamics of this resistant population, as well as the susceptible ones. Sometimes even micrograms and picograms of this particular antibiotic concentrations have been shown to be influencing the selection of this particular resistance in a particular population. So if the resistant bug was already there in that population, so given the concentration of the antibiotic and the structure of the bacterial population, the horizontal gene transfer seems to be predicted to take over in a large way. So which basically involves the transformation, the transduction, and the conjugation. So for example, it's been known for a long time that the type-4 secretion systems are involved in not only delivery of the proteins, but also in terms of the delivery of the DNA. In addition to that, they've been known to be involved in uptake of the DNA from the environment, so which basically is the transformation. And the type-4 secretion system is also involved in conjugation. And so this is especially true if the resistant pathogens are driven by the conjugative plasmids and the IS elements on the transposons, which leads to basically the conjugation process to rapidly go on within the population. If there is no resistant population within the bacterial subpopulation where it was supposed to be exposing to the antibiotics, then this leads to a lot of other dynamics which come into factors into consideration. For example, the rates and the magnitudes of the competing forces to select for or to select against the resistance. The fitness costs also come into the picture. There are a lot of de novo mutation rate, adoptive mutations, the fitness costs, the compensatory mutations, the genetic rearrangements, the physiology of the bacteria, the environment which drives this mutagenesis. All this become clear factors. So which indicates that the co-selection, so there's also the co-selection of the resistant markers and the epistasis. This also influences the distribution and the maintenance. So if there is no antibiotic exposure, but there is for a particular resistant gene, but because the other antibiotics are present within this particular pool, it is also possible that these conjugative plasmids, which basically encode for several resistant genes, continue to be thriving. As well, it is in these populations, it is also shown that the virulence gene seems to be associated with these plasmids. So if even the virulence is being selected out, so basically that leads to the resistance elements continuing within this population. So in general, this seems to be that the population dynamics and the resistance emergence as well as the maintenance involves several factors that leads to the resistance pool. But if you take what is happening within the clinical settings, so the clinical settings, one of the things you can think of is the microbiota, which is largely interacting with the invading pathogen. So what is known is that while they both are trying to look for a niche within the host, as soon as the antibiotic pool, the antibiotic concentration is sensed by both of these commensals as well as the invading pathogen, it seems to be that then they start basically going through conjugative process in order to lead to exchange of the genetic material. And in addition to that, some of these pathogens like Acinetobacter have evolved the type 6 secretion systems, which not only are involved in delivery of the proteins into the host, but also into the neighboring bacteria in order to kill them. For example, Acinetobacter continues to be like a predator towards E. coli, which continues to become a prey to it. So what happens is that because of the type 6 secretion systems, the E. coli is killed, and the genetic material of the E. coli is then taken up with the Acinetobacter. So this leads to a lot of exchange of the genetic material and intense horizontal gene transfer that leads to driving of the resistance within the pool. I hope this answers the question. Thank you.
Very well said, Chris. So we have one question from the audience. So she wrote to Ankur. So how about machine learning and artificial intelligence are useful to predict AMR? Ankur, could you please answer those questions?
Yeah, so I think that's a very relevant question. Machine learning is practically being applied to everything and anything these days as a matter of a solution-based system, and also as a matter of exploration. In AMR space, machine learning is currently being used in two ways. One is it is being applied to vast amount of hospital data and public health data that has been collected over the last few decades on AMR. And models are being built to which machine learning is now being applied to see how fast the resistance would develop to the new candidates that are coming, or the new antibiotics that are being introduced into the healthcare system. And those models are giving a reasonable level of prediction on how quickly a newly introduced antibiotic would end up becoming useless in a way, or obsolete for the system. So that's one way machine learning is being done. And the other way machine learning is being used is for tracking how the barcoding system, and when I say barcoding, this actually means the mutational signature of genomic clones that, as I previously said, are very, very good at picking antimicrobial resistance genes and these mutations with or without having any sort of selection. So these mutational signatures and these SNPs, as it is called in more technical terminology, is essentially providing a barcode, and that information is being put into, again, mathematical models to see how quickly these lineages would evolve to either become resistant, or how quickly these existing resistant lineages would evolve to become sensitive, so that we can plan the healthcare and the public health strategies better. So I hope that answers the question.
Yes, thanks, Ankur. So now we are moving to the next section. So priority list of antibiotic resistant bacteria. So when we talk about the antibiotic resistance, we majorly talk about the antibiotic resistance in pathogen, definitely because of its relevance. But recently we did some study when we isolated even the commensal bacteria from the human, and we did their genome sequence, and we could also identify that the commensal bacteria that reside in the human body, they also have enormous number of antibiotic resistant genes. And these antibiotic resistant genes are always linked with the monogenetic element. So now if the resistant genes is present in the commensal and resistant genes is present in the pathogen, so question is coming when we are going for the antibiotic resistant research, how to prioritize. How to prioritize which specific classes of bacteria should belong to the critical groups, which one should have the highest priority, which one should have the moderate priority. So we have two very eminent scientists and clinicians, Dr. Sayantan Banerjee, who was working previously with WHO, and Dr. Sulagna Basu, who is also working on the critical enteric pathogens in terms of the antibiotic resistance. So I would request Dr. Sayantan first to address how to prioritize antibiotic resistance bacteria for development of new antimicrobial and also antimicrobial resistance detection kits. Sayantan, please over to you.
Thank you, Dr. Bhavatosh, for this opportunity. See, before 2017, there was never a global effort to prioritize the antibiotic resistance pathogens or as a priority list per se. So there were published list of priority pathogens by CDC in 2013, and then by Health Protection Agency of Canada. But in 2017, for the first time, WHO commissioned an international review board to prioritize the most important pathogens for I would say advocating for more and more investigations and R&D and investment into research for antibiotics. So there were a few caveats in this priority pathogen list, which we should all know and discuss, and there are certainly certain strengths of it. Previously, till date, this global R&D activities, as Dr. Cole was telling, were mostly guided by factors like the perceived unmet medical needs and the investment pressure, because most of the new antibiotic and new molecule research was funded by the companies, pharmaceutical companies. So the market size, the availability of technologies, and the return of investment was a major guiding force for R&D investment for antimicrobials as well as any other pharmaceutical industries. Now, the major objective of this global priority pathogen list was to guide the prioritization of incentives and funding to help align the research and development priorities along with the public health priorities of the global need. So basically, first comes the understanding of the public health priorities related to AMR. Now, when this list was prepared, already mycobacteria, because in 2017, we all know that Mycobacterium tuberculosis, the resistance in Mycobacterium tuberculosis was already a big menace, and up to 20% of retreated cases were found to have different classes of resistance to fast-line anti-tubercular drugs. So basically, the mycobacterium, both tuberculosis and non-tubercular mycobacteria, were kept out of the global priority pathogen list. And for reasons which are unknown to me, fungal pathogens, parasitic pathogens, and viral pathogens were kept out of the list, probably because of giving more and more importance to the AMR bacteria. But around 2017, we did have significant reports of antibiotic resistance fungi, which should have, in my personal opinion, which should have come in the priority pathogen list, but that was not included. So basically what was done was a WHO commission for expert review, and a process was adopted which was known as multi-criteria decision analysis technique. In my multi-criteria decision analysis, basically expert opinion as well as evidence-based data are taken into consideration, so that the list of priority that we are making is complete, non-redundant, and non-overlapping. Okay, so what was done is, firstly, for prioritization exercise, there was a list of selection of a list was selected of antibiotic resistant bacteria. Okay, the list was quite long. It contained about 25 pathogens. Then there was a selection of criteria for prioritization. So how would we prioritize those bacteria that we have in the long list? Typically, there were a lot of criteria used, but mostly literature search was done for both peer-reviewed published literature as well as reports emerging from the countries and the regions through the GLASS platform that we have in WHO, the Global Antimicrobial Surveillance System platform. And primary importance is given to all-cause mortality. Next is the overall healthcare and community burden of those bacteria. And third was the prevalence of resistance in those bacteria. And fourth was the 10 years trends of resistance. These were the 4 criteria that were mostly used to prioritize the list of 25 pathogens that we had. Additional importance was given to transmissibility, to preventability in hospital and community settings, and to the treatability and current pipeline of antibiotics that were available. So based on this selection criteria of prioritized prioritized pathogens, data were extracted and synthesized, and scoring and weighting is done to finalize the ranking of pathogens. Now, what we need to understand, there was a brilliant software as we were talking about artificial intelligence and different integrated software models. There was a brilliant software which helped the prioritization list, the 1000 Minds initiative, which was used for the prioritization of all the criterion. And based on this, the bacteria were, the priority list of bacteria were divided into 3 groups: a critical group, a highly important group, and a medium importance group. Now, in the critical group, basically, we can't say today that we will disagree, because day-to-day medical practitioners like me or Dr. Roy will understand that in the ICUs, in the inpatient hospitals, we face the major brunt of these bacteria. In the critical group, there are the carbapenem-resistant Acinetobacter, the carbapenem-resistant Pseudomonas, and the carbapenem-resistant Enterobacteriaceae, plus the ESBL Enterobacteriaceae. So what we usually see is the greatest menace of carbapenem-resistant Enterobacteriaceae, Klebsiella E. coli, bloodstream infections, ventilator-associated infections, and catheter-related urinary tract, as well as catheter-related bloodstream infections.
Yeah. Dr. Banerjee, this is very interesting. Just as you are speaking about it, so what is the difference in AMR or the resistance patterns in India, and which clinical indications are more important from an India perspective rather than if you have to compare it from the world? Is the picture same, what we see clinical picture of the resistance same in India versus the rest of the world, or there is some differences?
So there are some differences.
I'm sorry to interrupt you. This I couldn't hold myself back.
This is a very important question, and we need to have a clear-cut discussion on this, because for us, we are using the CLSI breakpoint. We are using the UCAST breakpoint. Till date, we don't have an Indian breakpoint. Till date, we don't have Indian standard strains, and till date, we your question is very important, and it shows the limitation of my knowledge to answer your question, shows the lack of surveillance data that we have from India. Definitely, the major brunt in Indian setting is of the gram-negative bacteria, particularly when we talk about indoor inpatient settings and ICUs. We face probably because of the higher ambient temperature, because of the relative humidity, we face and I would say probably because of a major lack of good infection prevention and control system, we have a significant burden of gram-negative bacteria. So this problem is not typically, why we face MRSA problem less than gram-negative bacteria and critically ill patients is not very easily answerable by me. But in India, we do see a lot of MRSA in skin and soft tissue infections. But when it comes to, suppose, invasive infections like ventilator-associated pneumonia or catheter-associated bloodstream infection, definitely gram-negatives are far more important than MRSA. Similarly, when we talk about Clostridium difficile, C. diff is a very, very significant problem in the West, where in India, I have my own hypothesis, is that we probably are overflooded with so much gram-negative infections of Klebsiella and Acinetobacter, which grows easily. Probably we tend to overlook Clostridium difficile a bit in India. And the second hypothesis is that because of the moist and warm environment, probably the spores are germinate easier in the indoor facilities, and the vegetative form of Clostridium difficile is often killed by the while the transmission pathway. So in the West, we have a more significant problem of MRSA and Clostridium difficile, whereas we see more gram-negative problems here. But this answer is, I would say, it is not a foolproof answer. We have in the West nowadays the growth of carbapenem-resistant bacteria and cholesterol-resistant bacteria, and the problem of gram-negatives is hugely growing up. If you see the recent list of priority pathogens that are published from CDC USA in 2019, they have included fluoroquinolone-resistant Neisseria gonorrhea as one of the most critical pathogens.
How big is that problem in India?
So, practically, what I would say is our inability to culture urethral discharge is probably more important factor than the absence of gonococcus for underreporting in India. So, a vast majority of sexually transmitted infections, particularly urethral discharge, etc., and are addressed by untrained physicians, primary and secondary level physicians, and a lot of what I would say so-called sexologists who are not a part of the modern clinical microbiology lab associated modern hospital system. So
can I interrupt you for 1 minute?
Sure.
So particularly when you are talking that our inability to cultivate those gonococcal pathogen, but nowadays to detect a pathogen, you really don't need to cultivate. There are a lot of cultivation independent methods using that you can easily detect the gonococcal pathogen. So why people are not adopting that? What is the difference?
Yes. So majorly what I feel is the problem is in the translation. The science, the technology is available, but when we are applying that technology in the outpatient door settings, when we are applying the technology in the primary and secondary level healthcare setup, when we need to culture the cervical discharge of a lady, and a fresh sample is needed either for culture or NAT-based technology, which Dr. Bhabatosh is trying to focus on. The problem in India that we face is availability of the technology to the focal point where the treatment or the care seeking is actually taking place. So the major problem that we faced at maybe I can give an example of All India Institute of Medical Sciences, AIIMS. So we had a huge number of patients presenting with sepsis, but our blood culture positivity rates were low. A major reason for this is because most of the patients are coming through, are referred through various hospitals where they are getting a few doses of antibiotics. And when they present at AIIMS, often we collect the blood culture sample, when the patient has already received a cocktail of maybe 5-6 different antibiotics and our culture positivity rates are low. Okay, next thing is overall the pre-laboratory, the pre-analytical technique, the pre-analytical part of most of our government hospitals and most of our private, I would not say private hospitals because I'm not sure about the overall picture of private hospitals in India, but in most of the major public hospitals, the pre-analytical part and the modern availability of modern culture techniques are severely lacking. I can vouch for sure that in major medical colleges, state medical colleges across India, very few hospitals actually have a rapid blood culture system or a rapid fluid culture system, which includes, I'm talking about something like bactec or bacterial alert systems, which include antibiotic neutralizing resins and a rapid liquid culture system. Sadly, I have to tell this is when I was posted in Muzaffarpur during I think July 2019, when there was an outbreak of unknown hypoglycemic encephalopathy among children, and about thousands of children died within 1 month in Muzaffarpur. Some earlier researchers related it to lychee consumption, and because of the presence of hypoglycemia in lychee, the kids were dying. And we also found some other factors in it. When we were walking in Muzaffarpur, there were 3 big hospitals where patients were coming in, and majorly being referred to one big medical college in Muzaffarpur. And we didn't have a single CSF or blood culture system. So what I was trying to say is, I'm not wanting to pinpoint individual institutes, but what I'm trying to say is our lack of understanding, our lack of, what I would say, the culture of not sending cultures in India is so predominant that we don't have a robust, quality controlled surveillance system. That is a major problem.
Maybe in that sense, coming back to your earlier point in terms of the resistant Enterobacteriaceae. So if tomorrow there's a drug which tackles Enterobacteriaceae very good, so which clinical indication you will pick up in India? I'm more talking about India in terms of doing a clinical trial. Which is the major clinical indication for gram-resistant Enterobacteriaceae in India? Is it the hospital-acquired infections? Is it CUTI? What will you go for?
So for a new drug, particularly if you are talking about a parenteral antibiotic with a very wide spectrum of coverage, broad spectrum parenteral antibiotic with a wide volume of distribution, it depends upon the antibiotic that you are talking about. Okay, so I would definitely not. So if you are talking about something like nitrofurantoin, which typically is an oral drug, a new congener of nitrofurantoin like fosfomycin.
Oh, completely new, completely. Yeah.
No, what I'm trying to say is it depends upon your volume of distribution and the compartmentalization. If you're talking about a very broad spectrum antibiotic with a very high systemic distribution, then definitely I would like to try on particularly on 2 things. One is bloodstream infections in nosocomial setting, either adult or both on adult and neonatal sepsis, and second is ventilator associated or hospital associated pneumonia. These 2 would be my first 2 choices of trying a new drug against gram-negative bacteria. I would definitely want to invite a one-liner comment from Dr. Animesh Ray, who is regularly seeing this kind of patients in hospital. Dr. Ray, do you have any other indication or top indication? What would be your choice?
No, I agree with you, Shantan. So, I mean, the greatest killer in our wards, they are definitely ventilator-associated pneumonia. So that is one. And the bloodstream infection, again, they can be primary or secondary. So, catheter-associated UTI. So, I think that would also be a common indication for which this particular drug might be tried on for. But I agree with you. VaiBhaV nosocomial infection, they would be.
Yeah. So. Now, if I just quickly try to come back to my topic of priority pathogen list, then we can have discussion on the R&D initiatives. Is that, you know, for India, which I definitely feel, if we are talking about India in this VaiBhaV Summit, the focus is on our own country right now. What I feel is research and development of new molecules are definitely important, but the translation of research is equally important, number one. Number two is we are significantly lacking surveillance data. We are significantly lacking in culture sensitivity data. You know, when Dr. Gandra did his initial, you know, surveillance of hospital resistance data. Do you remember, Dr. Gandra? Initially, you had to restrict your work in the private hospitals, in the private labs, because of the risk of quality assured antimicrobial sensitivity testing protocols of our public hospitals across India. You know, I remember your phenomenal work with CDDEP and with Dr. Ramanan Lakshmana, and what I realize is, you know, till date, we need to understand that what is the pattern of antibiotic resistance that is the major burden of our countries. The problem in the hospital-associated microbiology laboratories are that they have a huge load of samples. They have multiple problems to tackle on. Now everybody is overloaded with COVID. At a time, there are hundreds of hepatitis and HIV samples which are needed to be tested. So there are multiple things. So often, quality antimicrobial susceptibility testing gets compromised in the medical colleges. We are not being able to follow, so most of the medical colleges are not being able to follow very good quality, get good quality of media, follow quality assured testing methodologies. Most of the medical colleges do not have MIC facilities. You know, even for colistin and other polymyxins, for whom this diffusion absolutely is not recommendable. Even for glycopeptide antibiotics like vancomycin or teicoplanin, for which this diffusion is not good. Till date, I can assure you that most of the medical colleges across India, we have to consider about the state medical colleges. We cannot, you know, think about the structure of India by seeing the All India Institute of Medical Science and PGI Chandigarh and JIPMER. Okay, so in most of the state medical colleges, facilities for MIC does not exist, and there is no repository. There is no national repository of antimicrobial resistant pathogens to find out what is actually going on. We have a huge
ICMR was trying to build that repository.
Yeah,
so
yeah.
there have been several attempts. I don't know.
So I was closely associated with the attempt of ICMR, and today is exactly 1 year, 1 month. 16th September was the inauguration of the National AMR Hub in Calcutta. So actually, the National AMR Hub was inaugurated in Calcutta last year with a lot of promise from the US ambassador who came with WHO India representative, ICMR Director General. But unfortunately, due to COVID-19 or due to other reasons, still we have this National AMR Hub yet to be opened. The issue is, if you see,
we have limited time, so
yeah, I'm sorry.
Discussion, but we have one relevant question from Dr. Rajkumar Haldar, who's the CEO of Urbanile Biomedical. What doctor want to know, what is the role of environment and climate change in the prevalence of priority pathogens in Indian context?
Yeah, so basically, environment per se, environment is closely linked with the selection of AMR, definitely. You know, when I went to my ancestral village in the district of Howrah in West Bengal, I was sitting with my uncles in the market, and there was one of my uncle has a agricultural, fertilizer shop, and I saw a 1 kilogram jar of colistin with a picture of a chicken, marketed as a chicken feed, chicken supplement for quickly enhancing the size. What I'm trying to say is, thankfully, we have been able to ban the use of colistin in poultry just last year. But what I'm trying to say is, in India, there are two significant reasons of environmental pressure on antimicrobial resistance. One is the indiscriminate use of antimicrobials in the animal husbandry as well as agricultural sectors. And number two is the indiscriminate effluence of the pharmaceutical industries and the hospitals getting mixed with the sewage lines, which ultimately goes to the rivers and sea, and often the water, the topsoil, is contaminated with antibiotics. So these two are the major environmental pressures of antibiotic resistance. And in environment, fluoroquinolones, polymyxins, and particularly in agriculture, azoles for containing fungus is being rampantly used, which has driven this major priority pathogens. One thing that I would like to add before ending is in the Indian priority pathogen list, which I dream to come up as a suggestion from this esteemed panelists, that we need an Indian priority pathogen list, because the priority pathogen list that we have is of 2017 and is of global nature, and particularly do not cover two very important things. One is what we are facing regularly in the ICUs and indoor wards is Candida auris and azole resistant Candida. So it is not a bacteria, but Candida auris is almost behaving like a bacteria. Number one, we need to have it as a AMR priority pathogen, not only bacteria. Number two is we need to include the cephalosporin resistant salmonella and overall salmonella typhi problem in the Indian priority pathogen list as a very high priority. So these two, I will stop by now, and I would
I think that's a great point, to be honest. Dr. Banerjee, that was fantastic from your side. It was lovely overview. I think what we need to do, if this meeting, if 3 hours we are spending here, can be a little bit more productive, we need to define what is the first and foremost. We need to define what are the most challenging pathogens from the Indian perspective, not copying WHO list on every other slide. That data across 3 or 4 major hospitals should somehow reflect the picture of the nation. That's one thing. And the second thing is what we need to know from a drug development perspective or from new antibiotic perspective, which are the clinical indications where mortality or patients are dying most because of the resistant bacteria. Is it the VAP? Is it the complicated UTIs? Is it bloodstream infections? Is it bacterial sepsis? Where are the number of patients which are dying because of the resistance, and which are the clinical indications? So these are the two main factors which we desperately need some data. Otherwise we are basically following what is published outside India, and AMR is more and more becoming an India-specific problem.
Thank you, Professor Kaul. It's really great suggestion. We can definitely include your suggestion in the minutes. So now, Dr.
Minutes kahan jayenge?
So it's have to send to DRDO. It's sent to DRDO.
DRDO?
Yeah.
DRDO wale kya karenge?
So they have been given the task of organizing. There are lots of different kinds of things involved in not only in biology but other sciences also, and DRDO was given the task of organizing the VaiBhaV Summit. So they will
I think I'm just trying to be looking at the fact, if we have to give really a proper recommendation, we have to make sure that it reaches the right set of people who can basically define a policy paper around in terms of what should be done.
Yeah, that's
and these 2 points which have come out of this are really, really important points. Means without this, we are not really fighting AMR.
so what we can do is, in addition to sending it to DRDO, we can also send it to the heads of the other funding agencies, and like ICMR, DBT, and CSIR, so that that knowledge is out there, that there is a need for these kind of things.
Maybe Animesh and Sayan can define the problem a little bit better. They can articulate it for you and put it in 1 or 2 paragraphs, which can be circulated further.
I'll be happy to do that.
Yeah, Dr. Banerjee?
I'll be happy
Dr.
to
Ray?
do that.
Well, I just want to make a comment. I think there are some answers for the questions, but I don't want to interrupt the flow. In terms of what pathogens we have, there is good data in India. And what is it different from India to other countries? We have some information as well. But I can speak now or later. I don't want to interrupt the flow. So I can talk about it later.
Okay. So now, Dr. Banerjee, it's clearly mentioned that what are the most priority pathogens in the so-called the WHO list. So in the top position is occupied definitely by the Acinetobacter baumannii, and also then it's very important Pseudomonas and carbapenem-resistant Enterobacteriaceae. So since Dr. Sulagna Basu is working on Acinetobacter baumannii, so I'll request Madam, if you could tell us, the audience, how Acinetobacter baumannii neutralize most of the antibiotics effect, and what are the different ways Acinetobacter baumannii also disseminate those resistant genes to the other bugs.
Thank you, Dr. Bhabatosh. So today I'll be, though you have asked me to concentrate on Acinetobacter, but I would also talk about another pathogen which is again on the priority pathogen list, which is Klebsiella. And while I do so, I will be talking about my experience from the studies that we do here at NICED on neonatal sepsis. So first of all, we all recognize that bacteria has a 3 billion year evolutionary advantage over humans. So it is absolutely, it's really difficult to outsmart them, and that's what we've been seeing through our studies and other global studies also. Now, talking about these priority pathogens, some of these pathogens, of course, have been considered as critical ones, as Dr. Sayantan has already said. So these are Klebsiella, carbapenem-resistant Klebsiella, carbapenem-resistant Acinetobacter, and of course, carbapenem-resistant Pseudomonas. Now, since we've been working on neonatal sepsis, one particular organism which we've seen to be consistently present across many years is Klebsiella pneumoniae. And recently, we've completed a study which is termed the Barnard study, where which was looking into the burden of antibiotic resistance in neonatal sepsis in developing countries. So the completion of the Barnard studies also came up with the same information, that is, Klebsiella is a pathogen which is seen as a major pathogen in neonatal sepsis. And this particular study was not only carried out in India, in one center in India, but they also included other centers from Bangladesh, from Pakistan, from Ethiopia, from Rwanda. So quite a number of low middle income countries were included here. Now, if you look into Acinetobacter, or if you look into Klebsiella, from the perspective of a microbiologist, I would really like to say or add something to what Dr. Ankur, Dr. Krishna has also added. So what we see in case of these two pathogens is the fact that they are able to acquire resistance very quickly. So their ability to acquire resistance and then again transmit on to other pathogens is immense. If you look in so particularly Klebsiella, if you see, Klebsiella has a number of different plasmids. The multi-drug resistant Klebsiella has a number of plasmids, different replicons are present there. And these replicon types in Klebsiella are mostly conjugative replicons. So they will move from one bacteria to the other. And these plasmids are large size. They not only carry carbapenem resistant genes, but they are also carrying a number of other genes along with them. So that creates a huge problem because one plasmid moves to the other bacteria, it creates resistance to a number of different antibiotics. So that is what you see in Klebsiella, and that is also a phenomenon that you see in case of Acinetobacter. Particularly, if you also look into the different IS elements, you will recognize that certain IS elements are like they are associated with certain genes. Like NDM, if you look, not only from Indian studies, different studies from Vellore, from our studies here, or from other studies in India, we have seen that NDM is primarily associated with the ISABA 125 element. So either you have a truncated element or you have a composite element, but you have this particular element always with NDM. Again, when you go into another particular carbapenem resistant gene, you will find that there is some other IS element which is generally associated. So this is a pattern that you see in these organisms. And this promiscuous nature of these organisms, of course, gives them an advantage because these can move. You will see the transmission of these genes across different species. The other thing which we have noted from our studies, also globally, also we have seen is that certain genes will have a kind of potential to persist. So you have bacteria that persists because Klebsiella is an organism present in the environment, in the human gut, and of course that is again a reason why it is also present in the nosocomial environment. So you have pathogens which have potential to persist in different environments, different niches. You also have certain genes which seem to be more potent to survive in different circumstances. Like, for example, we've been looking into antibiotic resistance for the last 15 years now. And since the prevalence of NDM, what we find is that NDM has been there in the unit since 2010. And it is still persisting as one of the most important carbapenem resistant genes in Klebsiella and in Acinetobacter. Now, and NDM is again another gene which you will find across different species. You will find in E. coli, Klebsiella, Citrobacter, Serratia, Acinetobacter, Pseudomonas. Now, take for example another particular gene which is the oxacelinase, the OXA48-like genes. The OXA48-like genes, again, are prevalent in Enterobacteriaceae, but you will not find them in Acinetobacter. In the same unit, in the same circumstances, you will find one particular gene which is moving, clinging on to different species, moving easily from one species to the other, whereas some other gene remains restricted in a particular species. So Acinetobacter, you will find a lot of the other oxacillinases like the OXA23, the OXA58, all of these. But you will also find NDM, but you will not find an OXA48 like gene, which is present in the Enterobacteriaceae. So NDM, of course, must be having some kind of an advantage probably because of its presence with the ISABA 125, or because NDM is moving in the F2K plasmids, which are present in which can move easily from one bacteria to the other. So there are number of factors here which makes a particular pathogen a critical pathogens, and it comes at the top of the WHO list. One other information which I would also like to share with you here is that if you look into the different variants of these genes, now 2010 NDM came in, you already have 29 variants of the gene. You have a number of variants of the KPC, a number of variants of the oxacillinases, and CTXM. If you look into the different variants of CTXM, it already has 224 variants. So what I feel is that bacteria also has understood that change is the only constant. So change is something that is absolutely necessary for them to stay ahead in the race. So NDM, if you will see, initially we had a lot of NDM ones. Now if you look into the global data, or if you look into the data from India, and a lot of data is coming in from China and some from Taiwan also, all of these show that NDM5, NDM7, they are slowly becoming predominant. The only reason I think is that the other variants are able to survive. NDM is a gene which is a metallobetalactamase, requires zinc as in its active site. So what we have seen is that NDM5, 7, or the others like NDM14, 15, all of these have the requirement of zinc is much lower. So these enzymes can even survive in a zinc depleted environment. So the requirement of zinc for these enzymes are much less. So bacteria have they are finding out ways to again keep ahead in this whole race. And so these are some reasons why I think the WHO critical, the 3 pathogens which have been enlisted as critical, we see the same pathogens primarily. If you talk about the neonatal infections, these are the same pathogens in most of the developing countries. So the list, of course, seems to be the WHO list, as Dr. Shayantan was also saying, and I would really like to add to that, is the fact that we, of course, need a good network of a surveillance network. And NICED is a ICMR organization, and ICMR has already come up with a national network. So this network itself probably will be extended. It already includes 6 nodal centers and around 16 regional centers. slowly it has plans to expand, but you need funds for that. And ICMR also has come up with the AMR hub, which was inaugurated at NICED last year. And NICED will be at the forefront of this fight against resistance. And we all hope that we can have our own priority pathogen list. We have our own set of breakpoints and all of that. But we of course need to come together and see to it that we can make it possible, because in this particular diverse country, the problems are innumerable and geographically very different. So we also work with some of the northeastern hospitals in Agartala, in Assam, and in Agartala particularly, we've been carrying out a study again on neonatal sepsis, and we find that their strains are very different from the strains that we see in Kolkata, primarily because Agartala shares its border with Bangladesh, and so they have a lot of influx. The border is porous, and you have strains which are very different to what we see in Kolkata here. So studies on the different sequence types of these organisms have also shown that though the genes are very similar, but the sequence types of these organisms are very different. So keeping in mind the heterogeneity, the difficulties, we hope that we can come up and do something about it. So, Dr. Bhabatosh, if
Yes.
I
Thank you, Dr. Basu. It's a really very informative.
Bhabatosh, I had one point. May I make it?
Sure, please go ahead.
Okay, so I'll be very quick here. So what is interesting is that in the last 50 to 60 years seems to be that these resistant genes, the virulence genes, and the genes which are required for survival in very toxic environments have all assembled into these conjugative plasmids and moving from one bacterial pathogen to the other very rapidly. If you look historically, most of the resistance used to be in the soil and the natural environment where there were no plasmids as such. So a lot of interaction used to be happening with the neighboring other bacterial species, though with antibiotics, probably because they were all natural. And to a large extent, a lot of these functions were residing within some of the biosynthetic pathway genes and loci. And over a period of time, so these were called as the producers. And because the producers were making antibiotic and they were supposed to be killing the non-producers which were in the vicinity, it turned out to be that the non-producers became much more rampant in terms of acquiring foreign genes in order to develop resistance. And it looks like when you look at by genomic analysis, the non-producers seems to be the ones who are the major culprits in terms of translocation of the resistant plasmids in mega sizes into the clinical isolates. So what you see today as clinical isolates is mainly coming from the non-producers of the antibiotics as compared to the producers of the antibiotics. It's an interesting observation, but the thing is it indicates that primarily after the setting of the antibiotic era and a rampant use of the antibiotics, the plasmids and the horizontal gene transfer seems to be taking over as compared to a stochastic mutation generations. I stop here. Thank you.
Thanks, Chris. So now I just go to Dr. Onimes. So we have some specific conversation. I remember that when I first met with Dr. Onimes, and I really thank Santon for connecting me with Onimes. So the first conversations I had with Dr. Onimes, and what he told me that any patients which coming to AIMS, we have capacity to treat. But if a patient coming to AIMS and if he is going to admitted to ICU, we don't know whether we'll be able to cure the ICU acquired infections. So Onimes, could you please tell us why these ICU and hospital environment are the richest source of extensively drug-resistant microbial pathogens. Whether it's only the uses of antibiotics or anything else could also contribute this emergence of those antibiotic resistance pathogen in the hospital environment.
Yeah, thank you for the question. So thank you for the question. A number of factors are responsible why, especially in the intensive care unit, we get a lot of antibiotic resistant organisms. One is, as you have said, that there's a lot of selection pressure because these patients who are very critically, they end up getting one antibiotic after the other. So there are also other host factors important. So these patients, most of them, they usually have, and first time they are being health. They have had higher exposure to antibiotics again. Them at a precarious for developing their antimicrobial resistance. Next is obviously the issue with catheters and central veins. So these catheter venous lines sit in the peak. Now they themselves they can introduce from the aged use infection.
I think we lost on MS because of the net connectivity. So, Dr. Saurabh, are you available?
Yeah, I'm available.
Okay. So
am I
on MS? We can't hear you.
Yeah. So, hello. Yeah. Can you hear me?
Now I can hear you.
Hello.
Yes, we can hear you, Animus.
I'm sorry. So I think the technique went offline. So long story short, so a number of host factors and a number of factors related to the which are found in the ICU lead to preponderance of resistance.
Hello.
Yes, Animus, I think.
Yeah.
Animus?
Yeah, I can hear you. I can hear you. I can hear you.
But we can't hear you. Your voice is totally breaking.
Yeah.
Okay, so we'll take you in the next part of the thing. So I will go to. So sort of that, for the antimicrobial resistance, one of the most important thing to detect the antimicrobial resistance in the pathogens. So the gold standard for detections of antimicrobial resistance is the culture assay. But there are bacteria, those are extremely difficult to cultivate. Even some of the bacteria, they grow very slowly. So that's why even before the resistance profile came to the clinician, they would not have any option other than to prescribe the antibiotic. So this is one of the major challenges. Till now, I don't think we have any rapid antimicrobial resistance detection for wide variety of pathogen. Most of the detection method is rely mostly on the nucleic acid based methods. But since you are working on the rapid detections of antimicrobial resistance based on the sensitivity or based on the new device, could you please tell us that what are the major limitations of developing those devices that can detect the presence of resistant genes without cultivating a box?
Yeah, sure. So I come from more from a diagnostic solution provider perspective. So it's very important for any diagnostic solution developer to understand the problem perspective. And I think it's very important to note that unless we know how to use antibiotics sensibly, we will grow resistance even to any new antimicrobials that come up. It's very important to know how to use it sensibly. And for that, it is important to have these rapid diagnostic tests in all settings, primary care and critical care setting. And this diagnostic development challenge, as you are alluding to, is multifarious. And there isn't a single user requirement case for the different kinds of problems that we have. So this is one of the biggest challenges. So for any solution developer, diagnostic test developer, needs to really understand the user requirement case, as I call it. What does the user require in that particular problem? So when we have thought about detecting antibiotic resistance, we have always thought about detecting the genes, but that is certainly not the definitive detection, because it is important for us to understand the phenotypic antibiotic sensitivity. And the challenges of quantitative detection of such phenotypic antibiotic sensitivity is quite high. First of all, there is so much of variation between the serotypes. So if we are going to detect, for example, based on an antibody or an aptamer, or say polymer imprinted polymer, there are so many different kinds of receptors which target epitopes on the bacterial surface. But there is so much of variation even within serotypes. It's very difficult to come up with a conserved target against which such receptors can be designed. And one idea could be to develop different receptors against different targets and have a cocktail of them. But then again, that adds to cost. And if we are targeting the primary care setting, which is the largest care setting in India or any developing economy, then we have to think about the cost perspective as well. So then I think there needs to be a research in identifying the common epitopes. And this is where diagnostic developers such as us, myself, coming more from an engineering physical science perspective, we need to go more into the biological aspects. And that is the multidisciplinarity challenge. It's important for us to cross our comfort zones and go to other disciplines and understand the problem. And in this case, I'm talking about understanding common epitopes. And then if we, for example, for a gram-negative bacteria, if we go to the core regions of the LPS, then it is possible to determine bacterial load regardless of what the serotype is, which may be very helpful in some cases, especially for working with sterile sample where the bacterial load is a big indicator. But it does not necessarily give the pathogenic load. It may be a pathogenic or a non-pathogenic bacteria. Again, if we go to sepsis, the problem is huge. Even though bacterial load is a good indicator in that case, but detecting, say, 10 bacteria within a billion RBCs is another challenge. So the challenges are huge. Again, when we come to another case, for example, diabetic foot ulcer leading to a chronic wound because of presence of bacterial biofilm, there, one idea is to detect the virulence factor, because detection of bacterial load means nothing there, because there can be a huge colonization there without causing an infection in particular. But then if we are talking about detection of toxins, then there are so many millions of serotypes that can be present. Each of them may be producing so many different toxins. One potential solution that one of the companies called WoundCheck has come up, for example, is to detect the protease activity rather than going and detecting the various toxins, which is, I think, one good idea to do. So To summarize, I think it's very important to come up with the user requirement specification, and that can vary for different problem perspective. But regardless of what the application is, regardless of what the user requirement specification is, one thing is always there as common. We need more and more of a digital readout. A digital readout which can then be wirelessly transferred into a portable device, which is AI enabled, and so that we can then integrate with the cloud to address this surveillance problem. Because we keep on hearing about the surveillance problem. The surveillance problem is because we are unable to gather data at the large scale in a cost-effective manner. So if we could address these core problems, I think we will be able to go close or near to solving this surveillance data problem. So to summarize, I would say that I would certainly love to collaborate with you people who understand the specific requirements of the various applications and work on that user requirements case and bring together a multidisciplinary team to come up with potential solutions for this. Thank you.
Thanks, Saurabh. So now it's very clear that from the history of these antibiotic uses, once you introduce the antibiotic, definitely the antibiotic resistance bugs will come. So, and it will depend how intensively you are using the antibiotic. So with that respect, definitely if we tackle to the antibiotic resistant genes, we need alternative of antibiotics. So when the bugs are coming extreme type resistance or multi-drug resistance, what are the alternative to antibiotics to fight with the resistant crisis? Since Dr. Manoj is working on the past therapy, so I think he is the best person to answer this question. So Dr. Manoj, could you please tell us what are the research going
Babatosh,
yeah.
Babatosh, I think we missed the topic in between.
No, global I kept in last. So once this is over, then we'll go to the global. So please, Manoj.
Yes, thank you, Dr. Babatosh. Yeah, we already hear this problem about antibiotic resistance, how they emerge and how the horizontal gene transfer play a huge significant role, and the selection pressure because of the rampant use of the antibiotics is causing the selection of all these antibiotic resistance bacteria. And we also hear a lot about what are the priority pathogens, especially the Klebsiella, Baumannii, Pseudomonas. These are the most one actually that we can actually, some of the bacteria that we can actually also culture. Obviously there are problems with the unculturable bacteria, but the pathogens that we can culture and we don't have a solution for those because of the fact that they are resistant to antibiotics. And also Dr. Anil Cole mentioned that after this pandemic, everybody's worried about how this antibiotic resistance crisis is going to come up because everybody is expecting that it's going to be a huge problem, which is because of the massive use of antibiotics in the current situation treating these COVID patients. And Dr. Basu also mentioned that how bacteria have been living with us and they have been evolving far more longer than us, and she said that we cannot outsmart them, and that's quite true. We all know this, but yes, we definitely need to seek alternatives, and there are definitely a lot of alternatives that are out there, either in the preclinical research area, or some of them have actually gone into the phase trials also. And some of the one that I think in the list that Dr. Babatosh sent me and wanted me to talk on about the antimicrobial peptides or even monoclonal antibodies. Yes, these are very nice alternatives, but I think the large problem with these is that although there are some of the monoclonal antibodies against I think with the C. diff and anthrax, they are already approved, but others are in the clinical trials. The problem with these are, I think, they're the shelf life of them is very poor, and once you have developed a antibody against one particular pathogen, and again you'll probably get a resistance again. That then again you'll have to think about making another variant of it, which is, I would not say it's not possible. It's possible, but it takes again a huge amount of money. But where is the marketplace for that is another concern. So they all have benefits, and also they all have these all disadvantages. And we want to go to that route or not, and the specificity is another concern. For instance, we always talk about using broad spectrum antibiotics, and now we have come to understand that when you use antibiotics and they are so broad spectrum, then they might will affect other normal microflora in your system, and the dysbiosis would occur, and that have another health problem. So we actually want, if you use antimicrobial peptides, you need to make sure that they are specific, but that is quite a challenging part. So there are all these other challenges with this, but I would actually want to focus on bacteriophages. Phages are everywhere. They are the most abundant organism in this planet, and they have been co-evolving with bacteria for as long as bacteria have been there. So there is a constant race going on between phage and bacteria all the time, and you can easily isolate phages from the environment with a very simple microbiological technique for any bacteria that you can cultivate. Now the metagenomics analysis is also suggesting that they are seeing the signature of this so-called viral dark matter, where there are phages against all these unculturable bacteria. Maybe there will be a day down the road that you'd be able to assemble those and somehow reboot those phages to target those uncultured, but that's a different issue. But at the moment, you can easily isolate bacteriophages against any drug resistance bacteria. We have been working in our lab targeting Klebsiella pneumonia, especially the carbapenem resistance bacteria, which was isolated from one of the outbreak here at the National Institute of Health that actually killed 10 different people, the cancer patients that were, and the bacteria was circulating around through the ventilators, and a lot of people died. And right now we are trying to address that by selecting a proper phase against that bacteria. But even with the phase as an alternative, bacteria also grow resistance against the phase. But even if the resistance emerges because of the utilization of phase, you can again go back into the environment and select another phase against the resistance bacteria. So there is an enormous source of this bacteriophages, in our environment that you can tap into and make utilization of it. But there is still challenges with it. There are a lot of challenges with it, even with utilization. It may sound simple, but still the challenges are. We talked about all this horizontal gene transfer and things like that. Phages are the one factor that do this transduction all the time, as we all know. So you have to be able to characterize these phase pretty well before you can actually utilize into a human patients. When you are utilizing a phase that is transducing, that has the ability to transduce, then you might be, getting into another problem by transducing resistance genes to the another bacteria. That's the other problem. And some of these phases, they carry toxin genes as well. And especially the temperate phases are the one, temperate phases are the one that go and integrate into the bacterial genome, and they pass along with the bacteria. And some of them, they carry toxin genes. And you want to avoid those kind of scenarios because that will be another problem. So for that reason, in the phage therapy field, when we say, utilization of phage, we always talk about isolating a good candidate, meaning that one, it has to be a lytic in nature, not that it would go and integrate into the bacterial genome. And the other that make sure that the phage genome these days, DNA sequencing is very rapidly done, and you can look through the gaze into the sequence and see that whether it harbors any toxin genes or not, or whether it has any other resistance genes or not. You can rule out those things and apply those into the for the therapeutic purpose. Now, as far as this phage therapy is one of the oldest technique, even before the antibiotic was invented, was used. And now that we are ushering into the pre-antibiotic era, so I think the phage therapy is gaining some momentum at the moment. And the phage therapy it has been quite successfully used in Eastern Europe and in Georgia, Tipsley, in Eliava Institute. Now it has also come to the west part of the world. Also here in the United States, there is an Center for Innovative Phage Application and Therapeutic Division in the University of San Diego, where the center was established based on one of the very successful story that happened was there was a professor from UCSD who was infected with Baumannii, drug resistance Baumannii, and he was literally under coma for so many days, so many months, for 3 months or something like that. And then they had no other alternatives to treat him, because every antibiotics they tried was not successful in treating this patient. And what happened at the end was they went into, they filed for an IND cases with the FDA and got approval for the compassionate use. And they applied bacteriophage against that particular bacteria, and he was able to survive. And during the course of treatment, he also developed a lot of resistance against those phages, and they had to re-purify, re-isolate and re-purify the new phage against those resistance bacteria. And ultimately, he was out of the wood and he survived. And recently, a nice report came from that center where they had treated 10 different patients. And the majority of the patients were suffering from either Pseudomonas and Pseudomonas staph, those bacteria. And but they had two unsuccessful outcome in that treatment. Was that especially patients who were suffering, who had a ventricular assisted device, had a lot of biofilm in that, and then the phage, although were susceptible to the bacteria in the in vitro setting, when they applied to those patients, it really didn't cure those conditions, probably because of the biofilm. However, but there is a huge research going on where you could actually engineer bacteriophage, for instance, to target some of these biofilm producing persisters, antibiotic resistance bacteria, such as there is an idea of, maybe engineer a bacteriophage with a high genetic payload so that it can produce some biofilm degrading enzymes, for instance, and can penetrate into such niches and take care of the bacteria. And there are also technologies that are emerging where CRISPR technology is being used, where the CRISPR-Cas systems are being integrated into the bacteria, such that the phage bacteria interaction is so fascinating. It's very specific. That's the one advantage of it. But at the same time, they are specific to the strain level, and sometimes it does not cover the whole strains of the same species. So that's one of the challenges there. But that challenge can be addressed by multiple different ways, either by using the cocktail of bacteriophages, where you would isolate a bunch of different phages against different bacteria and mix it into a cocktail. That's one approach to do it. Or the other approach is, such as the CRISPR technology, where even the phage would target one type of bacteria, but cannot have a vegetative cycle to go, but at least can deliver the DNA. In that cases, you could, engineer the phage to deliver the DNA with the CRISPR-Cas system just to go inside and knock out some of the resistance genes or some of the essential genes to destroy the bacteria. So there are all these different approaches that are emerging. But Dr. Babutos asked me on that list that are there any FDA-approved bacteriophages? At this moment, there are no FDA-approved bacteriophages. They are only being used in the compassionate care ways. I think it is going to go like this for a while. But although making these preparation of bacteriophages is not that hard, but it's very costly at the moment in a sense that you have to treat one patient at a time, and a lot of research is involved in it. And I think there has to be some sort of innovative idea to come up with, some sort of setting in a hospital where you have, a collection of library of good candidate of bacteriophages that you have already made a library of it. Then whenever the patient comes, you can quickly scan through those library and then probably supply phages to the bacteria. That would be more feasible if you have established.
So Manoj, what we are doing internally is we are engineering these phages with CRISPR-Cas. So we are introducing Cas9 in these bacteriophages, and there are a few other people who are doing across the world and trying to see, as compared to the native bacteriophages versus the engineered phages with the CRISPR systems inside, whether they have more cidal activity or not.
Yeah, I think if the bacteria is susceptible to that particular phase, whether you engineer or not, it makes no difference because it's gonna kill anyway, right?
I think in some cases, particularly in cystic fibrosis and bronchitis, where you have constant presence of Pseudomonas or other pathogens, the engineered phages tend to have more cidal activity versus the native phages. We don't know why, but apparently the engineering does kill more of the biofilm present phages as compared to the
I think we don't have a clear understanding of it for sure, but like I said, in the phase host interactions, a lot of the time it is possible that some of these bacteria becomes resistant with the native phages immediately, but not at the level of, for instance, some of the bacteria, they just set off their receptor and they don't even allow phage to inject their DNA. In that scenario, the CRISPR-Cas would be useless. But there are certain scenarios where it's the internal factor that actually inhibit the phase growth, like I mentioned in the vegetative cycle of the phase. In those cases, what happens is the native phase would not be allowed to kill that particular bacteria because the resistance is from the inside, that hinders one of the phase life cycle pathway. Then in that case, if the DNA is delivered and the CRISPR-Cas is there, at least the CRISPR-Cas will take care of it and destroy that. That's my feeling to it. That's why I meant to say that, that's why the CRISPR-Cas equipped one are probably better. Not only even that bacteria that has already evolved resistance to that native phase, but also some other strains of it, other strains of Pseudomonas that represent, that are not sensitive in killing by the native phase. That could be possible.
Yes, thanks, Manoj. So it's really very informative that most of the people had the idea that the native phage has been used to kill the bacteria, but the engineer part, particularly the CRISPR engineer, is definitely very new to most of the audience. So now we need to, because of the time constraint, we have to go to the next discussions, and our next discussion is on the global action plan on antimicrobial resistance. So before we go to the global action plan on antimicrobial resistance, first things we need to know what is the current mortality burden of multi-drug resistant bacterial pathogen in the Indian context, and then what are the global as well as national action plan and OMR. I think Dr. Suman Gandra is the best person in these panels who could give us very good insights. So Dr. Gandra, could you please enlighten us?
Sure. Thank you, Bhavdos. So I want to highlight a few things before we go into the mortality burden, which will be relevant to Dr. Cole's comments and some other people who brought into this. The first question is, do we have our own priority pathogen list? I think there is very good data from India to say that we have a list of pathogens that we should consider, and which aligns mostly with the WHO. Where is this coming from? The ICMR created a surveillance network which spans approximately about 25 hospitals, and the NCDC has its own network about approximately 24 hospitals, which are state medical colleges. So if you look at both data from these two networks, it's totally similar. And what we know is that the major pathogen in the community is E. coli, and especially the extended spectrum beta-lactamase producing. And from the hospital side, it is Klebsiella. So, which, and the third pathogen is Acinetobacter, which we already discussed. So there is rich amount of data for that to create our own pathogen list, and which closely aligns with the WHO. The second comment, how are we different from the rest of the world in terms of, what is different in our strains versus the other strains from the Western world? And how does it influence the pharmaceutical industry is about the resistance mechanisms. And as Dr. Basu mentioned, the most common resistance mechanisms that we encounter in our pathogens is NDM plus OXA. So actually it's combined now, both of them. But if you look at the Western world, it's all about KPC. So whatever the new drugs or novel antibiotics that are coming in pipeline are focused on the Western world. So none of them are active against the NDM as far as I know, which are approved already or which are in the preclinical phases. So that is India's specific need is to come up with drugs that are active against the NDM strains. And for running clinical trials, we have lots of patients. And with respect to India, the syndromes that we should be considered are basically from the community side, it's about pyelonephritis or complicated urinary tract infections, which especially includes the E. coli extended spectrum beta-lactamase. If we talk about nosocomial environment or hospital acquired, it's going to be ICU, ventilator-associated pneumonia, and also the bloodstream infections in the hospital setting. So that will be the real need for us. And we have lots of, the problem with doing with the novel drugs is the clinical trials, because many of the developed countries don't have enough patients to run even clinical trials. But that's not the case with India. So we have so many people, but I think it's about how do we safely conduct, in a strict regulated environment, how do you perform those trials? All right, so then the question about mortality. So we did publish on the mortality burden of multi-drug resistant organisms in India. And this is really a high-level data in terms of, we basically took a network of private hospital and looked at the outcomes in patients who have MDR, which is multi-drug resistant infections, versus those who don't have the multi-drug resistant infection. And what we see is the odds of death is about 5 times higher. And it's across, basically, even if it's ICU patient, the odds remains higher. Now, but what we don't know is about what we call attributable mortality. That means, is the death really attributed to the drug-resistant infections, or is it, something else that's happening, to the patient? So we don't have that information, and I'm not doing that research, but there's a lot of interest in to study the burden of multi-drug resistant infections, especially to understand the attributable mortality with that. So So we have good data from India at the high level, but we don't have the real attributable mortality information. But what we know is that we have very high prevalence of drug-resistant organisms based on the surveillance data that we have from India, from the ICMR network, and from the NCDC. So it is clear that we have high prevalence of resistance in organisms that are isolated from the cultures. And we have a ecological data saying that there's the odds of death is higher in multi-drug resistant organisms. So that's what I would say. In terms of India, I think the next question that you were asking, I'm just going to go back and see what was the question.
Dr. Gandra,
yeah.
Can I interrupt you here?
Sure. Yes.
I was wondering, as you said, NDM1, how prevalent, what's the percentage of Enterobacteriaceae which will be resistant to NDM1 in an Indian context as compared to the KPCs?
So with respect to India, I would say 98%.
Yeah.
I mean, Dr. Basu, you can correct me, but as far as I know, it's up to the higher 90 ranges.
Yeah, I think in the neonatal sector, neonatal infections, it's around, in Enterobacteriaceae, NDM is around 50%, whereas KPC is just sporadic. You will see them sometimes and then you just don't see them at all. For
Dr. Basu, does that mean that if you say 50%, does that mean that 50% of the patients who have these infections cannot be treated because there is no effective mechanism to cover the NDM resistance? Huh?
No, they can be treated, because even though you will find these bugs like Klebsiella or E. coli to be resistant to carbapenems, you will find that they are susceptible to an aminoglycoside like amikacin. A lot of the neonates are getting treated with amikacin, like in the particular units that I am associated with, they are getting treated with amikacin. Sometimes ofloxacin also has been, there is a lot of fluoroquinolone resistance, particularly in E. coli and Klebsiella. But even then, you will find that for ofloxacin, ofloxacin has been used in treatment of neonates. But so they are being treated sometimes. And what I feel is the unit that we work with, it's a tertiary care hospital, the IPGMER hospital in Kolkata. So empirically, they are using in their unit now, as of now, they are using Piptaz. So Piptaz is the empirical antibiotic they are using, because cephalosporin resistance is very high. It's like around 80-85% resistance in Enterobacteriaceae. So you cannot use, earlier they were using cefotaxime and amikacin, but now they have moved on to piperacillin tazobactam. So that's it. Yeah.
So I would like to add here. So yes, what are the current therapeutic regimens for the NDM strains is either colistin or any of those aminoglycosides, which are probably remain susceptible. So, but other than these, if you have and as you know, there's a lot of use of colistin resulting in colistin resistance. So once you're at that stage, we don't have many options, other than, again, if we are fortunate, we can have aminoglycosides. But the problem is, giving aminoglycoside is an issue. We are not using because it has its own side effects. So yeah. The idea is we want to come up with a novel drugs which are have less adverse effects and which target the strains that are relevant to India. So that's what I would say. And in terms of the proportion, I just want to, yeah.
Suman, just one question from my side. So even when you are talking the prevalence of NDM, so what actually we observed that even the prevalence in the same bacterial species, it could differ depending on the source of isolation. Right. So when you are isolating those same bugs from the sepsis patients, you find that the prevalence of NDM is quite high. But when we isolated same thing from the fecal sample of the diarrheal patient, we could hardly identify 1 to 3% of those bugs really carry the NDM. So it's not the bug. Also, the source of isolation, I think, is another important thing.
Yeah. I think I would like to add a little bit to this. Like we, while we were doing the Barnard study, the burden of antibiotic resistance in neonates, what we also we observed very similar. Like NDM is very prevalent in the bloodstream infections, but in the we were also doing the maternal and the neonatal gut samples. So there you find lot less. So what, and also we had carried out another study where we were looking into how long the gut, if there is an NDM, how long it persists in the gut. It does not persist very long. Once the neonate is discharged from the hospital and again comes back within a month, you will not find the bug, the NDM. So of course the antibiotic pressure is something which maintains NDM. That is very true. But Dr. Bhavatosh, what we found is that in maternal isolates, we haven't published the study, so I will not be able to go into the details of it now. But in the maternal fecal flora, there is NDM. So much less compared to a CTXM. Like CTXM is absolutely rampant in the fecal flora, but NDM is much less, but it is still there. For neonates, it's a little different.
So Dr. Boss, how do you explain it that the resistance prevalence is based on the source? Because the distribution of the drug and the isolate should be same across the whether it's the fecal or whether it's a bloodstream. What drives this? This is interesting.
Dr. Cole, this is what we were also like trying to think about. Why do we see this difference? We were looking into maternal flora. Now these mothers, some of them may have had antibiotic, had taken antibiotic during the like in the earlier stages of pregnancy, but probably they have the antibiotics that you have used for them is of course not a carbapenem, because it must have been a cephalosporin or a fluoroquinolone or something like that. So NDM does get selected in their gut isolates, probably because of another antibiotic resistant gene which is present along with NDM. So you will generally find these NDM isolates also have a number of the QNRs or the aminoglycoside resistant genes like RMTB, C, all of them are present in these isolates. So the mother may have been treated for a UTI, must have taken a fluoroquinolone, probably that must have enhanced the selection, but they have not taken a carbapenem. In case of neonates, these are neonates who have been in the hospital, preterm, low birth weight neonates who had a prolonged hospitalization. So these neonates, while in the hospital, were treated with piptaz, with a meropenem. So their gut is more, their isolates are getting selected, the meropenem resistant isolates are getting selected. For mothers, the selection procedure is a bit different. That is what I think is the reason why it is so. Again, if you look into MCR, like MCR also, I have seen very few studies from India who have worked on bloodstream infections and shown that MCR is present in bloodstream infections. Not too many. In our neonatal unit for the last few years, we have been screening for MCRs. We do not get a single one. We do not get a single one.
So that is consistent with the surveillance data as well. We don't see MCR in E. coli, at least in India.
So I have another one question. So it's also from one of our observation that the antibiotic resistance and the presence of antibiotic resistant gene sometimes we cannot correlate. Okay, if you look that some of the antibiotic that have never been used in human, but the bugs have almost like 80% of the bugs have those antibiotic resistant genes.
Right.
And second thing, what we observe that even the expression of those antibiotic resistant gene does not depend on the presence and absence of antibiotic. So when we did the proteomics of the multi-drug resistance bacteria in the presence and absence of antibiotic, we could find that the expressions or the level of protein in both the cases is almost same. There is no difference at all. Even when we look for the 34 antibiotic resistant genes, they could considerably express. So why, if the antibiotic is the only major driver, then why bugs should express those antibiotic resistant genes in the absence of antibiotic? There might be something else, other function they could also contribute.
Right.
What we have also found is in the similar lines that what we call intrinsic resistance and target-based resistance. So all what you have, NDMs, MCRs, and everything else is a very target-specific resistance. But what you also have, what we call efflux-based resistance mechanisms. Most of this with Bedeclin, we haven't found a single patient where we have a mutation in the ATP synthase gene, which is the target of Bedeclin. What we have found is most of the time, even patients who have not been exposed to Bedeclin, we find an efflux pump which gets upregulated and drives the increase in the MIC, though it doesn't lead to a clinical failure, but it does lead to increase in the MIC between 3 to 4 or 5-fold. So the resistance from a target perspective is completely different than the resistance which the bacteria, because of its environment, because of other drugs which come into play like clofazimine and other drugs basically induce similar efflux pumps. And these efflux pumps basically act as generic resistance drivers for several drugs. And it can be one efflux pump affecting the resistance of 4 or 5 drugs which are out there.
Right.
Absolutely, yeah.
So I also want to comment about this question, why you don't see in the gut versus, in the bloodstream. One potential explanation, again, correct me if, is about the way you acquire the infection. It could be that, many of these patients, correct me again, in even the neonatal ICU, they're here for a while and they have all these catheters in place, etc. So it's important to know how they acquire the infection. It could not be, it's a translocation, it may not be a translocation issue from the gut. It could be potentially that some healthcare workers are injected or having a catheter, what is the, whether the bacteria was present in the environment itself rather than the gut per se. So any comment, Dr. Basu, on that? I mean, I'm wondering if that's a part.
I think that could be a possibility. Like when we were doing this, when we were doing the Barnard study, there were a number of hospitals from different low middle income countries. Now, what we found was that in tertiary cares, the hospitals which were tertiary care had more of Acinetobacters, whereas the other hospitals where you do not have a level 3 intensive care unit, these do not have too much of Acinetobacters. So of course the intervention that you are doing, the amount of invasive procedures that you use do dispose towards
Okay. All right. So in that line, so one of the current study that I'm doing is basically trying to understand the source of bacteremia for the patients who have these multi-drug resistant organisms, and trying to understand whether these are preventable infections in first place. So hopefully we'll have some answers with respect to the type of organisms that we're talking about. But that's really probably one good explanation why you don't have the, Dr. Bhabatosh's comment about the resistance genes are different in the gut versus in the bloodstream, etc. All right, so I'm just going to go to the next point. Dr. Bhabatosh, I mean, I see in here is that how to optimize antimicrobial use in India's setting. I think, we did a systematic review, and what we know, most of the antibiotic prescription happens in the community. About 80% happens in the community and the 20% in the hospital. But what we understand is that every other patient who presents to primary care will get an antibiotic. So about 50% of the patients who are coming for any reason, they are prescribed an antibiotic. And so that is really a problem. And if you look into more closely, the inappropriate use, like overuse, it's difficult to quantify in settings like India, etc., because we don't have any good diagnostics, etc. So it's more of empirical-based treatment. But what we have seen from the data, we know that at least about that 50%, half of that is inappropriate. So there is approximately about 50% inappropriate use of antibiotics in the community. Now, with the hospitals, obviously, it's a difficult, when you have a serious patient and we have to give empiric therapy, it really depends. It seems the first line is most likely, as we talked, piptazo is probably the first line nowadays, and then if they have exposure, then they go to the carbapenems and then colistin, etc. It's a difficult task considering the type of pathogens that they're seeing in hospital environment. Any comments or any questions here? I can talk, but I just want to make sure before I go to the action plan, I want to get a sense about Bhabatosh, any specific questions for antibiotic use in humans or animals that I should comment on?
Yeah.
Sumant, if I may just ask one question here.
Yes, please, Saurabh, go ahead.
So you mentioned that 50% in 50% cases antibiotic is given, and out of that 50% is inappropriate. What are the key disease cases, for example, are these given? Are these related to respiratory tract infections primarily, or UTI?
So basically, in terms of the study that we published, it's about any all-comer, even if you do not disintegrate the data by infection syndrome, it's just an all-comer, 50%. But if you look at the infection syndrome, it's mainly the inappropriateness is mainly with respect to acute respiratory tract infection and diarrhea. So those are the two big conditions where inappropriate use is driven mostly. UTI, it's not inappropriate. Basically those are symptomatic urinary tract infections. You have to give antibiotics in that situation. The inappropriateness is basically what antibiotic you're giving, not whether to give or not. So most of the inappropriate happens with respect to acute respiratory tract infections and diarrhea. So those are the two major conditions why we give antibiotics in the primary care setting.
So again, if you could comment on the reason, because acute RTI is the antibiotic prescription is inappropriate, probably because there isn't a rapid diagnostic test which can infer on that. However, for diarrhea, in most cases, you do not even need an antibiotic. So isn't that more of an awareness problem?
Well, again, even for respiratory tract, most of the syndromes are viral, and and diarrhea as well is a viral etiology. So it's definitely lack of awareness in terms of, but the problem is not a lack of awareness. It's the way you practice, right? It's India practices is a customer-based practice, and then if you don't give any medicine, and they may go to a different person, or those are the other. There are social cultural issues that complicate this issue. Although physicians probably don't know that they don't need to give it, but certainly if they look as a customer and they demand sometimes, etc. So yes, it is an issue in both on the patient side, lack of awareness about when to have access, when to take antibiotics, and also from the physician side, and also there's a pharmacy, right? Over-the-counter prescription.
Yeah, this is a problem that we have known for a long time, but I think an evidence-based decision on prescription antibiotics would be good. Then the patient, the customer, would also know that this is the reason why I haven't been given antibiotic.
Right.
So yeah, for diarrhea, do you think there is any evidence that we can work on
in terms of when not to treat?
Yeah, with antibiotics.
Oh yeah, there's a huge amount of data on that. Yeah, if you just have diarrhea for 2 days and there's no blood or mucus, generally it's not required. So,
I think, yeah. So I just wanted to add there the question that Saurabh raised about diarrhea and use of antibiotics. And so we work very closely with the diarrheal center in Bangladesh that sees quite a lot of cholera and other ICDRB. And one of the main reasons why they actually do use antibiotics is for hospital management as well, because they have a limited number of beds. They have to turn around patients pretty quickly. And alongside that, it also helps in cutting down the transmission. If you kill those bugs, rather than just waiting for the bugs to by themselves disappear, it reduces. So it has some impact. So it's not that use of antibiotic in diarrheal cases is absolutely unwarranted. So.
So I can talk about the National Action Plan, Bhabatosh, is that okay?
Yes, Sumon, please.
All right, okay. There are 6 pillars on the National Action Plan. One of them is improving awareness. The second one is strengthening knowledge through surveillance in terms of how much the burden we have. And the third one is about reducing the infections through infection control or in the community sanitation, etc. And then optimizing the use of antibiotics. And the last 2 are, investment into research and development and strengthening international collaboration. So I'll just walk through. I want to make sure I remind some of the major steps taken by India with respect to AMR. I think one flagship that I would say is the AMR surveillance network. I think there's no doubt that was a very, very good program and successful run through the ICMR, and now it's NCDC as well. So, and I think we should really congratulate the effort that they have taken. And of course, there's a lot of room for improvement, but I think that's one of the biggest achievements, in my opinion, so far that has been done in India, with respect to AMR. The second issue is about the fixed dose combination antibiotics, and they were banned in September 2018. And not all of them, but at least there was a significant amount of drugs, including antibiotics, they were banned. And what we see in the data is that yes, it actually works, and many of the fixed dose combinations that were banned are not actually sold. So that's really a very good move from the government side. And the third aspect is about ban of colistin for growth promotion. I think that's a really an important step with respect to India, and I think that's something to congratulate. But there's again more room to talk about in general, decrease the use of antibiotics for growth promotion in animal sector, and the next one is about the recent one is about coming up with the pharma levels, in the effluents, the minimum level of antibiotics that should be in the effluents coming from the industries. So this was basically a notification, but certainly that's a really first time that any country has produced in terms of these are the norms of antibiotics in the effluents. So it's a very good move, but I think the biggest issue would be about implementation of that, and how do we go by where the technology is not up to date in many of the industry sector. So, but still, I think that's a really good work. So if you go by each aspect of the National Action Plan, in terms of awareness, has India done good in terms of awareness? I think the only thing that I can think about is the Red Line campaign, which was done a couple of years ago. But I think nobody knows what happened with that Red Line campaign. And it seems it's no longer red line, it's a red box now, which again, not many people are even aware what red box on antibiotic is. So I don't think there's a lot of awareness created, at least among the general public, with respect to the topic of antimicrobial resistance. So there is a lot of room for improvement in that, in my opinion. And in terms of the surveillance, I think they have done a very good job with respect to the ICMR surveillance network, and within their capacity, this is really good. The next steps will be to how to expand to the regional and beyond, sub-regional centers. So this is the best one, in my opinion, they have done so far. But what they're lacking is the surveillance in the animal side or the environmental side. So that's going to be a huge task. And certainly there is an initiative from the animal side that they're doing surveillance from the Agricultural Research Institute, I guess. But that has not yet produced any data, but that has been started. In terms of the infection prevention, certainly there were guidance documents produced by the NCDC and also the ICMR for the hospitals. That has been a good step, but the question would be, how hospitals are implementing that is going to be a challenge, and the other aspect is going to be environmental sanitation in terms of preventing infections. How could we provide better sanitation? So that's going to be a huge challenge again from implementation side. And the last one, the other one is optimizing the use of antimicrobials in human side. The problem for India is, they don't have a surveillance of antimicrobial use, even in the human side. And it's going to be really, again, challenging, considering that we have 70% healthcare happens in the private sector, and the data is not out of the government side. But there are very good databases to monitor antimicrobial use, because there are companies who have sales information. So there's a way to monitor antimicrobial use in India through sales data that can be acquired from the private companies. And if you only look at the public sector, the use is about 5 to 10%. So there's not much antimicrobial use in the public sector. So a lot to improve. I think there's a large need to understand antimicrobial use surveillance in India. And unfortunately, I think this step, the government, as far as I know, they haven't taken many steps in this direction yet. And in terms of the animal side, again, the only good news is about the colistin ban, but certainly it's more important about whether we can stop the use of antimicrobials for growth promotion across the animal sector. So if that would be ideal, but certainly it's not easy, I guess. As we already talked about the pharmacy effluents, and they recognize, especially DBT, recognize this as a major problem in terms of and easy to control. And they did come up with the standards, but as I alluded earlier, the important challenge would be, how do you implement this? So that's a big question. In terms of investments into the AMR research and development, I don't know. Obviously, DBT is one of the organizations, and I think it's in diagnostics or drug discovery should be from the DBT side. So I don't want to comment there. I don't know much about what's happening. And the last is about the international collaboration. So certainly there's the government is working with the US CDC in terms of improving infection control practices in hospitals, or at least surveillance of hospital-acquired infections, and also antimicrobial stewardship. So there are measures taken and working. But the big challenge for the country is, all you can do from the central side is to give states directions, but it's up to the states who have to take at the ground implementation, and that's what it's lacking. As far as I know, only 3 states have come up with a action plan to implement the one produced by the government of India. So those are the things that I all I tried to give was an update about what's happening in India, as far as I know, and where there's room for improvement. And I'll stop there and happy to answer any questions.
I think, Sumanji, it was very nice. It was well summarized. I think you made very effective points in terms of the pharmaceutical effluents going into the rivers, and that is really a huge impact in terms of the APIs manufacturing, which happens a lot in big cities like Hyderabad and other cities. So that's a issue which needs immediate redressal. I think the hospital surveillance, as a lot of hospitals, particularly if I look at PGI, where I was a little bit associated with, and as a part of the National Antimicrobial Task Force, there was a specific mention in terms of building up those hospital surveillance, microbial resistance, monitoring those resistance, and also the proper use in, at least in the major hospitals. So there's a database which gets collected, but as you rightly pointed out, private sector is 70% of the antibiotic consumption happens in the private sector. How do we monitor that? I think in terms of the progress, in terms of the awareness and communication of misuse of antibiotics, that's something where we need to work a little bit more. But people, I think the COVID has come as an opportunity in terms of the general awareness and pushing this general awareness more now towards the AMR. People at least understand that there's a viral infection, it has a huge impact, there's a bacterial infection. So the infection control measures can be more strengthened, or people will be more cautious about infections, susceptibilities, the impact on healthy lives, cleanliness. So these are things which have come as an opportunity. So the National AMR Task Force, we need to monitor that. I think there is already data that antibiotic use has gone down in the last 3 months because of the COVID. So, so there is some kind of that the misuse which was happening is somewhat have been reduced. So I think there are several things which you rightly pointed out and messaged it up very nicely. A lot of progress has been made. And people, these are the challenge is that these are inter-departmental implementations like animal husbandry, the science and technology ministry, the health ministry. There are so many different players in the whole scenario that need to be aligned. And but I think there is some progress at different levels. But the key is that, once again, coming to the fundamental question that as we go ahead, how do we discover new antibiotics? How do we support antibiotic research and development? How do we implement strategies for proper use of antibiotics? Even if you have a new antibiotic, it may be lost in 5 years, because if you let it be misused, there is no mechanism you will be ever saved. If a new antibiotic is pushed in the first line treatment and not as a third line or the fourth line regimen, the way we have seen in the HIV drugs have been fantastically managed because they have been defined as first line, second line, third line, fourth line treatment. And I think we need to do the same thing with the antibiotics that have the proper access, but at the same time proper control in terms of the right markets, the right indications. And eventually, I think the last thing which I want to talk about was that we really need to identify, maybe you have the data, but I think we need to identify what are the India-specific issues in the AMR. Is the NDM a biggest challenge which we need to address as a nation, as a country? Do we need to screen the drugs against the NDM resistant strains as priority, or the KPC resistance or Enterobacteriaceae resistance in general is good enough to cover India as a population? So what are the key resistant hotspots across different pathogens that need to be taken care as we discover and develop new drugs? So, but overall, I think, thanks for the summary. I will just stop here now.
So I just want to make a comment, Dr. Cole, about the impact of COVID and antibiotic use in India. You know, this is what we're looking into. I have the data. I'm looking into that. It's not what we're thinking. The antibiotic use has skyrocketed, unfortunately. The time when it went down was lockdown period. That's all, because there was no access to medical care. So I think there are some lessons learned with respect to COVID, how the awareness can be used. How do we prevent infections? So that way we can reduce the use for antibiotics. But unfortunately, the COVID itself has led to inappropriate antibiotic use because of the initial reports of, azithromycin being
maybe the azithromycin class, but the rest of the classes must not be used so frequently. Maybe you will see more azithromycin resistance coming through, but not the rest of the antibiotics.
No, including azithro, amoxicillin, clavulanic acid, doxycycline, everything went up. So, but anyway, but I just want to give a comment there. All right, I'm done.
Thank you. Last question from this audience. So you are talking that use of antibiotics in the poultry has been banned, but now a lot of processed food, they use a lot of antibiotics. So is there any specific regulation how to stop the use of antibiotic in the processed foods?
So as far as I don't know, but again, I just want to clarify the use of the ban was only for colistin. I'm not talking about any other antibiotic. And that was also for growth promotion in a prefix feed, a premix feed. So that doesn't mean that a farmer can still give the antibiotic as a metaphylaxis. What means that if there's one chicken that is sick, you can still give it in your water supply and then feed all the chicken. So the question, I just want to clarify the person who raised the question. It's not in poultry. The only thing that was banned is the colistin, not other antibiotics. So number one. And in terms of processed food, as far as I know, no, there is no regulation as of now. The only thing I know is that they came up with a regulation for the antibiotic withdrawal period from the safety authority from India, food safety authority, is about there should be a minimal time period of antibiotic withdrawal period when we have the food. But that also, there are so many reports, even after coming up with that, there are several studies or reports that talked about high levels of antibiotics. So as far as I know, there is no regulation for even food processing.
Okay, thank you, Suman. Since we are out of time, so I'll request Dr. Nair to give the closing remarks.
Can I make one quick remark, short remark, before Deepak starts?
Sure.
But there is a very fundamental thing that we miss in this. There is somehow this belief that government and government institutes and ICMR and DBT can solve the problems. It will never be economically sustainable. We need to get our startups involved. In fact, many of among the startups that are doing some of the best antimicrobial research in the world, about 50% of them are in India. Some of them are molecules that are in clinical trials. Very little support from the government of India other than giving a 50 lakh grant. We need to get, it has to be economically sustainable, economically viable. We need to get startups. There is not a single startup person in this. I don't think government can solve all our problems. We need to get industry involved. An Indian startup is really good seen now. So it's very unfortunate that we didn't talk about that. But I just want to put that out there for people to know.
Thank you, Rams, for the last comment. So we, at least we did try to invite people from NCDC and ICMR who participate in the AMR surveillance, but they were not available at the time this was scheduled, so they could not attend. But overall, it's been a very illuminating and enlightening last 3 hours, and it's great to hear from people who are associated with AMR in different aspects of AMR, and it's been meaning we have a better idea. I'm sure all of us have a better idea about all different aspects of AMR. Thank you for so much for taking the time out and talking about different issues that Bhabatosh has so very nicely listed, and he had organized the discussion very well. And thank you everybody for participating, and also people who are listening in from YouTube. Thank you for your questions and thank you for participating. So with this, we can end this discussion. But I think it might be a good idea, since we are all connected through email now, to continue some of this discussion over email and keep in touch about different aspects of EMR. And we will try to generate a report. There will be a report for DRDO, but maybe we can also try to generate a report which we can circulate among the panelists, and they can add and enhance it, add to and enhance and refine it, which we can then send to the heads of different funding agencies and even to the Ministry of Health. With that, Bhabtosh and I thank you for your participation and for all your inputs. And depending on where you are, good afternoon and good night.
Thank you. Good night. Bye-bye.
Thanks, everyone. Thank you.
Thank you. Thank you.
Thank you.